Yo, yo, yo. What is up? Beautiful people. Well, some of you gotta be beautiful, right? What are the odds?
Oh, some of you gotta be
At least one of you.
right? What are the
Hold up.
odds? Oh, some of
My
you.
echo. Alright, let me see what's going on here. Got my microphone. You know, it's one of them ones. It's gonna be one of them ones. Late night. I was just, you know?
Well, I was feeling like streaming, man. I don't know what to say. Tomorrow's gonna be I got a lot to do. I'm hyping myself up. New year, new me. Got to be letting down less people next year, getting more things done. But till then, yo, here we are in the building, the hardest working. What's up, JP Jeffries? How's it going? I'm an animal. It's kind of rude. I think I'm a civilized human being. What's up, Bob Bravo? Bob said, I'm here, sire. Yo, chill, man. Yo, we're not starting a cult. Come on. Come on. What's up? What's up? Louis said, dang, I thought I was going to go to bed. I'm excited for my drive for a fish in the AM or replay action. Yeah, all you degenerate fish shop owners. Yeah, we're going to have at least a little replay for you in the morning as you question your life decisions. Slanging fish, okay? Happy New Year. What's up, Sage Cave of Wonders in the chat? Here we go. Here we go. Another stream. Off to a great start already with some fantastic people in the chat. Got a not too much on the agenda for this one. I just felt like talking, so I'm gonna make it your problem. Uh, and I need to set up this little fry tray because we're having a good issue of having enough eggs from things that I need more places to put them. And I really just not about that, like, I don't know, tub thing or whatever. We're only doing shallow rimless fried trays. Okay, that's it. 1.5 shallows. It's the only fried trays we're using. You can keep your Section 8 fried trays. All right. This is like no mixed income housing for the eggs. This is all pure rimless stuff. So I gotta set. What's wrong with me? I moved around some things. I made some space, and we got to set up this. This is going to be the next one over here. So I got to get to that. Where do you get yours? I happen to get mine from Elite Menagerie Habitats. You know, just a store that I have recently started becoming a show for. But yeah, that's what I've been buying them from. Louise brought some to the swap. I copped another one. I'm going to have to do it again sometime soon. That's the thinnest layer of substrate. Well, it's for hatching eggs, bro. It's for hatching eggs. You got me dying. No Section 8 fresh. Hey, yo, I shouldn't laugh too hard. I might need one of those joints sometime soon. But yeah, no, we got the premium fried trays. Everything about them has been the best experience for me, and I don't care what anyone says. It's what I choose to do. My racks, my choice, and that's what we got going on. So I got to set up one because my freaking Aeneas, the green Aeneas, I've been complaining about them not laying eggs. Well, guess what? They laid some eggs, so I had to put them in another one. Then we got the gold laser quarries. Now, I did have a decent amount of some gold laser. I found a decent amount of dead gold laser fry. I feel like I have heard people talk about quarry fry having a decent attrition rate. I don't know. There's still a lot of gold laser fry, but I did move a bunch of them to the yellow shrimp tank 5-gallon that is often here. I gotta say, man, nothing beats the aesthetic of looking into the distance and just seeing my rimless fry trays right there. Okay, those are the other two that I got. Now my dream, I'm conflicted. I like being able to look through the sides at them, but okay, I am considering this table over here. I think could perfectly fit 4 of them all together on this table. I think I could fit 4 of them all side by side, and then really I could see from above just fine. So I really wouldn't need to go from the sides that much. And actually, the sides would be so clear, I probably could see through each one of them to the next. So I'm considering that I might do that. I really might. There's one of the hongs away from Big Steve. He's been Big Steve is definitely someone. Yo, he be breeding so many fish. He needs to start. He needs to start wholesaling. They'll stores hit up Big Steve quick before the next one gets all of his whiptails. I think I had to swap like 5 different people asked to wholesale whiptails to him. It's pretty hilarious. When Nick starts a saltwater take, I'll know the world is ending. I mean, bro, you need to check the news. Like, aren't we bombing Iran or something next? Like, come on, bro. It's not gonna be my fish take that does it. Okay, okay, things are brewing. Now, what else we got going on here? I want to talk about or read about. We did Boost the other day. Boost is nice. It's cool. It's kind of a high tier plant. I was like, what's a bit more of a common plant? I started thinking about dwarf sage, Alcinaria. What are some papers on those? There's got to be some extra info on those. Some weird insights, perhaps, that we can learn from academic writings. So I might take a look at that. This tank has really been improving, man. This is the one that's been green behind me for so long. It's come a long way. I think throughout this year, I've kept adding to the panda cory population. I think it's at over 20 panda cories. I do take them to swaps, some of them, and then maybe I'll take like a group to a swap, and then for every group I take to the swap, I probably save like 1 or 2 pandas for myself. So I've ended up with a decent amount. They spawn pretty regularly. I just need to clearly. This is a situation where many eggs are going to be consumed. I mean, it is what it is, right? It is what it is. Oh shit, we got the Fish Fam Link bot in the chat. What's up, Fish Fam Link bro bot? How's it going? Sage says it's interesting the generational differences between fish keepers. I watch the old guys. What kind of differences you notice? And I like to think I take much inspiration from the old guys. It's a lot of old fish keepers that I'm constantly asking for help. Those are some nice metallic guppies. What kind are they? They're nice. I think the rainbows in there have been nipping their long fins, so I'm probably gonna take those rainbows out. But these are, I actually don't know what he calls them, but I got these from when I went to Ross's place last. He gave me a few when they were fry. There were some coals he didn't like as much, and you can see some of their fins have gotten all beaten up. Like that guy's fin's been decimated. So I'll probably be moving those rainbows. I meant to do that by now. Maybe I'll do it on the stream. Oh look at them panda cores. They're going at it. They're going at it. Look at them. Yeah, this is turning into a fun tank. It's nice actually being able to. I'm fresh. That kind of makes it sound like you want to eat me, dog. Like, yo, this is some fresh catch of the sea. Yo. Yeah, I don't know what kind of guppies there are. You have to ask Ross. But he's got some nice ones. He's got some nice ones. I'm definitely going to be trying to breed these. So that's one reason why the rainbows also would need to come out. I was thinking a guppy and panda cory tank is what this would be. I'm putting on shoes just in case I splash some water because I think I'm just going to go ahead and move these rainbows. I'm thinking of it. I guess technically it shouldn't take that long. And shit, let's do it. Hold up. Hold up. But I had it's been kind of a decent, I've been a little bit like under the weather the last few days. A little bit. I know it's just hard to tell because it's just my boundless energy, but a little bit under the weather. I'm not getting all the stuff done necessarily I'd like to. Not the day before yesterday. Whenever I did the stream that is not currently public, I think I slept like 16 hours at least. Like I was I was complete toast. I just could not get up. And I didn't like stay up late or anything like that. I was just totally knocked out. My legs were like numb and all chilly and stuff, and so I've not had the most energy to do too much. Okay, well that was easy. I think I'm putting them in right here. Yeah, we're gonna have to get some cuttings because I need to. The tank I'm putting them in, I need to add some plants too. Give me 1 second. What's up, buddy? What's up? Where are you going? Where are you going? Where are you going? I need to move the mop anyway. It keeps floating to the middle, blocking my view. Come on. Come on. Where you at? But yeah, but things are kind of like, it kind of feels like things are ramping up. Like if I ride the wave into 2026, I get a little bit more productive. I finish editing some of these videos. I get some work done non-fish wise, and we kind of keep this rolling. I think it's going to be a fun year. As far as breeding goes, it's kind of closing the year off uh, at a pace that I kind of enjoy, where stuff is breeding. We're not slaughtering things at a high pace. Fish are staying alive, I would say. And some of the stuff that's breeding is like cool shit, man. You know? So I really can't complain too much. Well, actually, no, I can't complain. My life is a hell. But I can't complain that much. Things are on an upward trend overall, I would say. I'm repotting my stuff. Oh, what the? A big ass fish just jumped. The hell? Yo, one of these cribs somehow just jumped out. Not cribs, sorry. What the? Okay, give me a second. How did you jump out, bro? It's these damn John said these were jumpers. Um they're not pelvicochromis, they're the God, I don't even know why I'm blanking on the name. Probably because I'm trying to get this guy off the ground. But that's why wearing these overhead headphones sometimes I gotta make sure I keep like an ear off because you need to be able to hear what the hell is going on in the fish room, bro. Else you're not gonna hear when that fish jumps out the goddamn tank. Look what just happened, bro. What were you thinking? Don't do that again. You must have done that right when I opened this lid. It's crazy. What you repotting, a couple of fish? I saw you say you repotting some shit. Make sure I got this lid back on tight. Okay. Fish off the ground, crisis averted. Fish catching these colds. We should be catching these fish. My life is hell. Same man. Same. Yeah. Yeah, man. Every day I wake up, I'm like I'm living in hell. But okay. Give me one second. Wash my hands. I touched the fish a little bit. Sorry about that. All right. Repotting stuff was a great time to talk about some plants then, eh? That's what I say. I say it's a great time to talk about plants. So let me pull up these papers I was planning on discussing. I was planting on discussing. Easy Sweetie said, what up, what up? Finishing up in the fish room. I'm about to I'm gonna do read some about some Valsnaria. I think we're gonna do big macro lens, big camera picture today. I'm liking how some of the fish are looking. I think maybe I might actually impromptu work on making this tank look a little nicer since I already got started on that a little bit just by catching those rainbows out. And I need to move those trimmings below anyway. But yeah, I got the Panda Cory army. So next year, I'm hoping to breed a lot of Panda Cories. Right now, I kind of just breed one here, one there. You know what I mean? I have a lot of ideas. We'll see if I can survive at a high enough capacity to execute any of them. But right now, yo, I gotta turn the camera on the newts. I'm sorry, pandas. These newts are just, they're nuts, bro. What's wrong with them? What is wrong with these damn newts? Give me a sec. Set up the camera. Yeah. I did work on the new. Yeah, that's good. We just escape some quote unquote. We'll improve the look of some takes. I actually worked on that quite a bit today, removing some duckweed and things like that. Like you actually might notice this tank has significantly less algae than it has in previous iterations. Redid fry table just finishing up. That I'm gonna be doing the fry thing soon. That's my next shallow over in the distance. I'm gonna make into a fry hatching thing. I'm telling you, bro, the shallows they're the best, man. Best fry trays. I'll never go back. I'll never go back. Never. I'll keep Louise in business just by buying rimless tanks over and over again if I have to. It's not out of the goodness of my heart. It's because I got a problem. I got a problem, and I feel like these tanks might solve it. It's in my soul. So the thing about these newts, it's like watching Wiley the Coyote. Okay, all I see in here is more and more shrimp all the time. I always see more and more shrimp, and I see them continuously whiff at trying to eat the shrimp over and over again. Like he's coming up on this shrimp right now, and the shrimp is like, "Bro, you're literally so slow. Like you're a joke." Now, half the time the shrimp don't even bother moving before the newt gets to them, but I guess this guy is particularly hungry right now, so I'll put in some pellets and see if the other newts come out. Couple fish sets, potting some abuse. I've been neglecting and potting up stuff I haven't put in permanent pots. Permanent pots. Yeah, I don't know why. That kind of sounds like a jail sentence. I'm in this pot forever. Really, dog? That's crazy. That's actually crazy. Yeah, okay. Let me get this shit up on the screen. Hold up. So I was kind of realizing I like know nothing about Vallisneria and all those kind of plants, like absolutely nothing. Um, I don't know what's going on with them. So, hello? Why is it putting up this screen? It's like not on the right. It's not on the right fucking tab. Wait a second. But I kind of want to learn more. I also want to figure out more about the difference between them and like the subulata and things like that. Like, there's just a lot of questions I have about these grass like plants. Also, I'm really trying. I've been so lazy on this, but guys, like, I have to stop saying and like so much when I'm thinking. Like, I feel like I used to be better at that, but recently I've been realizing I'm just relying on these nonsense words a lot. Like, I just need to pause, So, keep me up on the practice of that. So we got some invasive hybrid Valsnaria situation going on. That's kind of crazy. Uh, we got some systemic. See, I just fucking did it. We got some systemics of Valsnaria. Okay, that could be interesting. Molecular phylogenetic analysis of Valsnaria spiralis, Linnaeus, and Kaniaka Kamari, Tamil Nadu, India. Yeah, this one looks like it's highly readable. So I like that. This looks kind of like a short little thing. This invasive hybrid thing, though, is probably what I'm going to start with. We got submerged. 1 second. This thing is too big. It's covering up my beautiful newts. Okay. We can't do that. Cannot do that. It's not cool. All right. There we go. Okay. Submersed macrophytes, Vallisneria natans, and Vallisneria spinulosa improve water quality and affect microbial communities in sediment and water columns. I thought that was kind of interesting. That definitely sounded interesting to me. And then what's some other ones? And I got to pull up the subulata ones. Oh yeah, Vallisneria, novel species, taxonomic revisions, and hybridizations. Let's go. I think plants got to be one of the things where with fish, it's obvious to me that some people are going to be like, no, don't hybridize the fish. With plants, it seems like there's like some cool plant hybrid stuff that like is like chill, right? And that's fine. Maybe it's because some of the plants, like the way that you got to hybridize them is like it's not as likely to spread in nature and ruin things. Perhaps it's one part of it, but I have noticed that seems to be treated a little bit differently. So let's start with this invasive eelgrass hybrid Vallisneria pseudosolata in southeastern United States. Depends on which type of plant and who you ask, TBH. Okay. So there is a hybrid of Vallisneria densurulata and Vallisneria spiralis that apparently is an invasive species. And this is a Wikipedia photo of Vallisneria densurulata. Okay. And then let's see what this other spiralis was the other one. Spiralis, is that corkscrew? Just assuming. I don't know if there's more than one. This kind of looks different than the corkscrew that I have. Corkscrew Val. Okay, yeah, this is Vallisneria contortionist. Interesting. I guess that's a contortionist is a bit more creative than a corkscrew. So I guess I appreciate that. I appreciate the originality. Let me turn the brightness up on this. Yeah, I just did it again. Once I notice it and I call myself out for it, I'll trade myself. We'll get rid of this. But apparently, this Vallisneria ex pseudo or solata. So I guess that's because it's the spiralis versus the other one that has solata in it, kind of. So they're calling it Vallisneria ex pseudo or solata. It's an invasive aquatic weed that has recently become a major issue within the U.S. Southeast. It's a hybrid between two non-native eel grass species, Vallisneria spiralis L and Vallisneria densurulata machino, and has rapidly overtaken water bodies in Tennessee, Alabama, and Florida. Yo, they got the carpets, bro. A couple of fish says X notates the hybrid of the parent species. Okay. Apparently, they got the carpets growing like crazy out there, man. This is how you got to get your tanks. You got to get your tanks like these invasive weeds. This hybrid can reproduce rapidly through offshoot formation and floating propagules capable of drifting large distances before establishing. So they can float. And a propagule looks like a vegetative structure that can become detached from a plant and give rise to a new plant. Okay, so they're really spelling it out for idiots like me. So invasive is my favorite plant word. It means it'll grow fast for me. Jesus. So they got these floating propagules, which I guess is a word for literally the plant being able to float and then propagate elsewhere, capable of drifting large distances before establishing. Has been previously found in Japan and is thought to have been introduced in the United States by the aquarium trade or through dumping. Yo. So Vallisneria L is a genus of submerged macrophytes that have often been a major target for phytoremediation and restoration efforts. So now I've got to look up macrophytes just to make sure I understand what we're talking about here. Okay, science direct. Macrophytes are literally quotation mark large plants, where some interpret to include large filamentous algae, bryophytes, and liverworts, as well as higher plants. For the purpose of the chapter, we will define macrophytes more narrowly as vascular plants in which specialized cells transport water and minerals from true roots. Interesting. Okay, so we got some broader definitions, some minor, including angiosperms, ferns, mosses, bryophytes, and macroscopic algae that grow in or near water bodies and can be free-floating, submerged, or emergent. They play a significant role in aquatic ecosystems by influencing physiochemical conditions, biogeochemical cycles, and community structures. Okay, community tank. Hold up. But what if? So, a genus of submerged macrophytes that have often been a major target for phytoremediation and restoration efforts. Often, American eelgrass, or Valsneria americana is a primary species for restoration efforts in the United States due to their natural habitat, forage, and structure it can provide to many ecosystems. However, there's been a steady decline of the species throughout many aquatic systems. Simultaneously, research has determined that there are two native species of Valsneria in the United States, V. americana and Valsnerio neotropicalis, VICT, southern southern eelgrass. Most of the current research in Valsneria has focused on how to restore V. americana to landscapes and reasons why it may be disappearing. However, there's a sudden and rapid invasion by short-stemmed eelgrass, VX pseudoroslata, which has received increasing attention in the last 5 years. It's thought to have been introduced from aquarium trade as an ornamental Valsneria plant for sale, but was later dumped into a water body and has spread rapidly through the U.S. Southeast. It's also been thought to have been used in native Valsneria restoration under the guise of being a native hybrid. Both parental taxa of V. x pseudorosata, V. spiralis, and V. denistrata are non-native to the United States and originate from Eurasian continent. Valsneria x pseudorosata has infested the Tennessee Valley Authority system, the TVA. This population is thought to have been established sometime between 2018 and 2019, so it has moved quite fast. The introduction to the TVA system is not an isolated incident. It's been found in multiple systems in Florida, Alabama, Tennessee, and recently Mississippi. All right, so we know where to find the free, fast-growing Valsneria. It's time to go on the collecting trip. Of course, they have confirmed it in Florida with one site in Alabama. There's little understanding about its interaction with other organisms. However, there's anecdotal evidence that's rapidly displacing the invasive species Hydrilla verticillata. Interesting. So this invasive species is displacing another invasive species, Hydrilla, which is considered one of the most. Oh, this looks like some kind of like an Acarus type plant almost. Yeah, this looks kind of like an Acarus situation. I could see how it could be one. If it grows like the Acarus in my tanks, man, there's nothing I have that grows like that shit. I mean, low tech tanks and this stuff could fill up a 20 long in a week. It's absolutely ridiculous. Okay, it's incredibly clear that it can rapidly become the dominant plant within a water body. It is hypothesized that it grows as an evergreen perennial and can form large floating mass of propagules, upwards of 10 miles, which has led to its spread. The propagules are capable of floating for long stretches, and maybe why Mississippi was recently invaded. So they think this shit is just draining through the TVA system. It's connected. The TVA system flows into the Mobile River and ultimately drains into the Gulf of Mexico at Mobile Bay. All right. Morphologically, many Valsneria species are incredibly similar with few diagnostic characteristics. This crypticity equally applies to this plant, which can impede early detection. It's kind of like if they're all so similar and stuff, I'm kind of curious, like what could be different about this invasive one that is like a problem besides the fact, okay, I get it, it's out competing a native species, that's bad, whatever. But like if it can serve the same function, I mean, is it a total issue? I don't know. I don't know. I guess maybe we'll find out. Often this hybrid can be identified by, oops, I just totally lost my place. Low key, I'm rooting for the invasive plants dog. Dog. This cross is unlikely to have occurred naturally as both parent species are completely disjunct. Since it's been distributed widely through the aquarium trade under the names Vis viralis, Vallisneria rockstar, and potentially other common names. Okay, so they think this is an aquarium creation, 100%. I see. So, how you can tell what it is, it can be identified by a bright green leuconal band running up the mid vein to the leaf tip. However, this can be difficult to determine on young leaves or if water clarity is an issue. Another characteristic, and the most diagnostic, is a partially lignified stem that it will grow just above the soil line. This stem structure was described by Waskura et al. In 2016 that grows on mature plants, but not every individual has shown this in the field. This stem can be 1 to 12 centimeters in length and typically has a small but apparent internodes with adventitious roots growing from the node. So if we go to figure 1, okay, go to figure 1 here, and in A, we see the typical growth form as a rosette with long, thin leaves. So that's it over here. I'll make this slightly bigger on this side. Okay. And then B, asexual reproduction usually takes this form of stolons that extend from the rosette base. So here we got the stolons. C, when established, stolons with many small ramets can extend more than 30 centimeters. So we got, I feel like we kind of just see them as like runners, little runners here. And then here we're going to see the bright green leuconal band that follows the mid vein in C. So it looks like this right here. I do kind of see a bright green band here. And okay, then E, leaf tips are obtusely angled and appear rounded. So we can see, yeah, here, that is kind of a rounded tip. Pause. Then F, serrations are small, dense, and along leaf margins. So you can't even really see it from this zoom level, but on F, you can see the you can see the serrations here pretty clearly. Steve said, "Hey yo, what's up, Big Steve?" Then G, we've got the stem formation during experiment in mesocosms. H, we got chains of ramets in both still and moving water. And then we have the pistillate flower, which are the only flower type currently found. So these are the little flowers of this plant. What's up, Zebra Aquatics? How's it going? Yeah, I can see how this thing could spread quite fast. Look at that. Holy. All right, so this invasive aquarium hybrid. Ugh, it disgusts me. Disgusts me. Many species produce runners that have been observed growing above and below the soil line, often with upward of 10 to 20 daughter plants. Anecdotally, these runners have been observed detaching from the parent plant and floating long distances before settling in the sediment again. Vallisneria or dioecious. Now I'm looking up another damn word again. Here we go. Dioecious of a plant or invertebrate animal having the male and female reproductive organs in separate individuals. That's what that means. That's apparently what these plants have. But this hybrid has only been observed with pistillate flowers, leading researchers to hypothesize it does not reproduce sexually. So looking up pistillate again so I can remember. Okay. Yeah, got it. Right. A plant having pistils but no stamens. So I believe that is totally making sense to me. They only got one of the parts on each one. And Nick, did you see 13% of my octopus? Did you post it on YouTube or you send me a picture? Because I haven't checked my phone in a little bit. If you sent me on my phone, then I wouldn't have seen it. Preliminary observation has shown that it can handle low to freezing temperatures. Let's go. Today I learned that Nick is dioecious. Bro. Okay, I have not seen it on YouTube, but I'll have to check it out. I'm going to check it out after this. But the entire genus has presented multiple complexities for systematists, taxonomists, and evolutionary biologists. Particular species like ribbonweed, Barosneria australis, and V. neotropicalis are still debated in many circles, often getting lumped under the name of a congener. Aaron said, "Did you see my octopus? Pause." Hey, yo, man. He's trying to show his tentacles in the chat. That's crazy. It's crazy, man. Just as crazy. Watch out, man. Watch out. Man, watch out. I'm going back to the pandacories. Pandacories are too lit right now. They're too lit right now. My pandas bring all the girls to the yard and they're like, man, all this shit look the same to me. I'm not gonna lie. I'm not gonna lie. So here we got a comparison between this Vallisneria hybrid and the native Vallisneria americana and Vallisneria neotropicalis. Okay. So an A on the left is the hybrid, and then B, we have the hybrid americana in the center and neotropicalis on the right. They said both Americana and neotropical leaves can have different colorations based on growing depth, but may be typically darker than these other leaves. So you can see it is quite a bright green compared to the native vals, which if this was an aquarium hybrid, maybe that was their motivation. I don't know. I don't know if this one's more dwarf than ours or something, but a brighter green color. I can see that being marketable. But you can definitely see a distinct color difference. A is a comparison of left and center. Here they are in the view. So this is the hybrid on the left. This is Americana, and this is the Neotropicalis one over here. So I would say the hybrid does. I mean, look, they all fucking look pretty close to the same thing, but if this is a consistent color difference that they have, then I would say it does look significantly different. I do think it's interesting that it's mixed with spiralis, so I guess it probably has a little bit of twisting in it. Zebra asked Steve about the octopus if he's tried at night with a red light. And Steve said, I've tried with the lights out and without a flashlight. Red light never crossed my mind. He definitely doesn't like the bright light. And he's been trying to coax him out with food. He's got feed 3 times. I've yet to see him, so he's winning. Yo, bro's got an octopus that he can't see. It's crazy. And it's only going to live a year. Should have listened to that store employee, man. He said they make terrible pets. So far, how's a tropical employee is up 1. Steve is up 0. Oh, Steve, there's one of your hongsloi, man. There's one of your hongsloi. You're always asking if they're still alive. Look, I haven't killed them yet. Watch out. Man, watch out. Okay. Here is a map displaying the known locations of this invasive species. So, premium hongsloi. Yeah, no, luxury. This is a luxury hongsloi for a luxury streamer. Okay. Hit up Big Steve. I hear he's got hongsloi coming out his ass. Pause. The Tennessee is just getting eaten up, man. They're getting eaten up by this hybrid. Look, boom, boom, boom. It's all up this damn TVA. Man, Florida's got a bunch, but bro, it's Florida, dog. Like, come on, they got everything there. I'm just gonna assume, like, as soon as the next wave of COVID, maybe the bubonic plague or something happens, like, I know Florida's gonna be one of the early adopters of that shit, man. It's a filthy ass state. Invasives everywhere. The current observations tied with how little is invariable. See, they keep saying it has great concern for further spread, and its rate of invasion of concern spread is a major concern. But I want to know what the concern is, because it looks like they have not actually studied the ecological impacts yet. So I get that they're concerned, but like, I did hear someone make one compelling argument about like invasives being natural, or like it being a natural thing that stuff from other places somehow ends up, like, birds carry all sorts of things very far. And you know I mean, obviously that's like an extreme, but sometimes I kind of do feel that way. You know what I mean? Like, whatever, man. you know like, sometimes I do kind of feel with certain invasive stuff, I'm kind of like, whatever. This one is one of those. I'm like, I need it. The floating plants. A lot of the invasive floating plants is pretty easy. Just, they've done a lot more to emphasize why they're so damaging. They need to up the speed on this and let us know why we need to be scared of this hybrid Valsnaria. But let's look at something positive regarding Valsnaria. Here is the submerged macrophytes, Valsnaria natans and Valsnaria spinulosa, improve water quality and affect microbial communities in sediment and water columns. Now, one of the reasons I'm kind of looking at this, I'm like, maybe I'm sleeping on Vallisneria as just a good cover for breeding stuff. Like, I've kind of poo-pooed it a bit. I mean, just other stuff you could carpet that are probably good, like, obviously Swasser Tang and things like that. But shit, it's easy to carpet some of the dwarf sag and stuff. So, I mean, why not just commit to it and just, give in? Dwarf Chainsword is another. I'm about to have so much Dwarf Chainsword. Bob says the 10 invasive geckos that visit my house every night, and Steve said Bob has geckos for sale. That's funny. Yo, you know what we should deep dive, bro? We should deep dive vampire crabs. On God. On God, no cap. What's wrong with me? Yo, I'm on one tonight, bro. I'll say anything. Healthy aquatic ecosystems are essential for human beings. However, anthropogenic activity severely worsen water quality. In this study, using assembling mesocosms, we've developed an efficient and easy-to-handle method to monitor the water quality, measuring the electrical conductivity of water. Ooh, the EC. Our data demonstrate that the growth of two submerged macrophytes, Vallisneria natans and Vallisneria spinulosa, improves water quality by decreasing the electrical conductivity. Furthermore, using the high throughput DNA sequencing, we now analyze the microbial community abundance and structure in the sediment and water columns with or without plant growth. We generated 33,775 amplicon sequence variants from 69 samples of 4 sediment groups. The results show that the relative abundance of bacteria was higher in the sediment than in the water column. Moreover, the diversity and composition of microbes biomes were altered by Vallisneria sp growth, and the diversity of the microbial communities decreased due to the submerged macrophytes in both the sediment and water columns. The B diversity of the microbial communities also varied significantly with or without Vallisneria growth for both the sediment and the water columns. All right, let's go. Let's go. Okay, I'm gearing up for this. I hope we're going to get to this reading. Phytoremediation through aquatic macrophytes is a green and economic strategy that has proven promising for restoring water quality. Aquatic macrophytes are macroscopic flora that can be separated into 4 groups. 1. Emergent macrophytes, 2. Floating lead macrophytes, and 3. Free-floating macrophytes, and 4. Submerged or submersed macrophytes. Submerged macrophytes, as essential maintainers of low trophic levels, are essential in water ecosystems and crucial in nutrient recycling and promoting ecosystem biodiversity and self-clarification. Self-clarification, bro. Correct itself. The earth is healing, thus reconstructing their health. Submerged macrophytes stabilize freshwater ecosystems, promote the removal of pollution from the water environment, and improve water quality by one, physically absorbing the pollutants due to their large surface area, two, absorbing to support their growth, three, absorbing and degrading pollutants, and four, providing an adherent surface for microbial community and producing adherent biofilms. Submerged plants and epiphytic biofilms provide an appropriate microenvironment for microbiomes, thereby removing pollution. It gets deeper. I'm sure. Yo, every time I pause, I'm just gonna say it gets deeper. Like this is just some grand conspiracy right now. The physical, chemical, and biological components of aquatic ecosystems exhibit an intimate interdependence. Submerged macrophytes and interacting microorganisms constitute the main components of the self-purification abilities of water bodies. They help the water bodies self-purify. Aquatic plants not only generate oxygen through stem and leaf photosynthesis, creating an oxygen-rich environment in the water, but also recruit the microbial community with strong metabolic ability for organic and inorganic matter by providing carbon sources and other matter to support the microorganisms through secretion from stems, leaves, and roots. Microorganisms provide carbon dioxide, secondary metabolites to host plants, and even inhibit the growth of harmful microorganisms, thereby promoting plant growth. Many studies have demonstrated that microbes and submerged macrophytes interact to improve water quality. Microbial diversity affects water quality and is also affected by it. Thus, microbial diversity can be a bioindicator in aquatic ecosystems. Many studies have recently focused on microbes in sediments and epiphytic biofilms of urban black odorous ecosystems. Urban black odorous ecosystems, yo, I just got hit with a microaggression in a Valsnaria paper right now, dog. yo, I'm about to call my union representative. Okay, get this paper writer, man. He just called me a urban black odorous ecosystem. It's just crazy. As well as other aquatic ecosystems that contain submerged macrophytes. What's up, Mick? Mick said no sleep for me this night again. But Mick, that's your fault, dog. Take a fucking melatonin or something, okay? Some sleepy time tea. You gotta sleep, man. You gotta sleep. As a matter of fact, Mick, someone was saying they were like, "Yo, that Mick guy in your chat, he is so interesting. He's so fascinating." I was like, "Yo, shut up, bro. He's just a fish nerd that never sleeps." They're like, "No, I'm telling you, this Mick guy, he's cool." I'm like, "All right." They're like, "You gotta get him on the stream or so." Look, Mick, I want we should do a stream. I wanna have a conversation with Mick. Let's do the Mick interview, okay? You let me know. Steve said this guy is so boring I can't keep my eyes open. Dog, you already can't keep your eyes open. You're falling asleep because you've been waiting so goddamn long to see your octopus, but it's never gonna happen. You keep staying up till 3 a.m., 2 a.m., 4 a.m., just shining all your little toy lights at the tank, okay? And it's not, yes, Mick, we gotta do a stream, dog. We gotta, I wanna do the Mick interview. I do. So let me know when you're free. You don't gotta show your face if you don't want to, but yeah, we gotta talk, man. If anything, we gotta talk about your sleep schedule and how we can make some progress here. However, most studies performed in natural water bodies may be affected by many known/unknown factors, and the microbial diversity affected by submerged macrophyte growth under laboratory conditions is rarely reported. Valsnaria sp are important submerged macrophytes that are widely used to improve the water quality of fresh ecosystems and have attracted increasing attention in recent years. So that's why I was finding so many damn papers of these things. When was this written? This is the February 2024, so that's relatively recent. Yo, see Mick? Steve said, "Hell yeah, let's meet Mick M." I'm telling you, bro. Yo, this guy is fast. He's a gym. He knows every single fish. He knows about all these other critters beyond fish. Like I think we have a lot to talk about, honestly. Now, we'll see how coherent it is to talk to someone that only sleeps 30 minutes a day. Like we might find that he speaks out loud in a different language. You know what I mean? But yeah, no, we gotta talk to Mick. We gotta meet this guy. Honestly, guy, it could be an alien for all I know. Yo, that's why you don't gotta sleep. You probably just plug into the hive mind and they didn't give him the extra mental boost to make it through. Valsnaria important? Yes, yes. Okay, so Valsnaria, I guess, are like big in the rehabilitating urban black odorous ecosystems world. Like they're hoping they're gonna come and clean things up. Nick said one day, one day. All right, well, keep us in mind, man. We want to chat. Whew, Valsnaria natans and Valsnaria spinulosa are two species of the genus Valsnaria with similar morphologies. Spinulosa is thought to be endemic to China. It is a dominant submerged macrophyte in lakes of the middle lower reaches of the Yangtze River, often coexisting with V. natans. I haven't heard a single thing negative thing about this invasive owl so far. Well, we're on to the next one. We're not on the invasive owls anymore, but I do think that they didn't convince us that it was evil beyond the fact that it was taking over. But like the Americana, I guess, just needs to get good or something. Often coexisting with venatans, while venatans is a cosmopolitan species, and I know I've looked this up before, but what's a cosmopolitan species? Let's just cosmopolitan distribution. Animals with cosmopolitan distribution are those whose range extends across all or most of the world in appropriate habitats. Okay. In this study, we conducted mesocosm experiments to detect change in water quality. And mesocosm is any outdoor or indoor experimental system that examines natural environment under controlled conditions. In this way, mesocosm studies provide a link between field surveys and highly controlled laboratory experiments. And Bob says, let it grow, let it grow. All right. They use some DNA sequencing. They analyze the microbial community abundance and structure and sediments and water column and etc. Materials and methods. Finally, that last one was just like a think piece. Give me the experiment. All right. Materials and methods. This is the best part, honestly. I think this is the best part when we do these things. Seeing how they set up these damn experiments. 9 plastic tanks, 250 liters diameter, 80 centimeters, and height 52 centimeters. Okay, so 31 inches by 20 inches. Okay, were used for this comparative experiment. Each tank was filled with 20 centimeter layer of soil as sediment and 30 centimeter of water. The soil used in the study was taken from the top 0 to 10 centimeter of soil from the Lushan Botanical Garden, Poyang Lake Branch Garden, located in Weijiatown, Zhujiang City, Jingxi Province in China. We get the longitude and latitude if you're into that kind of a thing. They collected the V. natans and V. spinulosa from the Changxi Lake, a shallow sub-lake of the Poyang Lake, the Jingxi province. The seedlings were generated from the seeds and winter bundles of them, and 10 centimeter high seedlings were planted in the tanks, with 20 plants in each of the 9 plastic tanks. They divided these 9 plastic tanks into 3 groups. One had no plants. One had V. natans, and the third had V. spinulosa. Each group concluded 3 replicates. So they got the control with no plants, and then they have a tub, 3 tubs with each plant, and then a duplicate. So 18 tubs. No, wait, 3 replicates. So they got 27 tubs. All tanks were exposed to natural sunlight outside the lab with little shading. The light intensity ranged from approximately 5,000 to 90,000 lux. All the tanks were periodically watered with tap water to maintain the initial water level. By the end of the experiment, the final leaf lengths of the plants were approximately 50 and 30 centimeters for V. natans and V. spinulosa, respectively. Each 30 centimeters about 1 foot, each 15 centimeters about 6 inches. Learned this from reptile terrariums. Okay, I'm going to try and remember that. Okay, electrical conductivity. The electrical conductivity of the water columns was measured using a portable HQ 4200 pH/EC/TDS meter. Let's lift this bad boy up. Mick said Hongsley is preparing to spawn that tank. I do think they have been looking like they're up to no good. And I've been feeding a bit more worms in that tank and some higher protein foods for that purpose. And I did put the camera on this tank just because now that it's not green, you can see I actually have a nice little group of panda cories in there, and they're usually quite active and out and about. Now that I have about 20 of them, so there's always something going on in this tank. Okay, some shoes pulled up, but here we go. So if you want to have a dope little meter for TDS, you want to perform flexible pH, DO, and multiple parameter tests with dual probes and reliable IP67 build. This is going to run you $2,190, and they couldn't even make it look cool. This thing looks ancient. It looks like a fucking Palm Pilot. It looks disgusting, honestly. Like, bro, you had to make it look like a toy. Yeah, I'm not rocking with this. We need to get Apple in, okay? We need to get Apple in. We got to make the IP67 that looks sexier than this thing. I'm dropping 2 bands. Yo, can we throw a Louis Vuitton logo on this thing or something, dog? Let's see what it does. Equipped with 2 probe inputs, the HQ 4200 allows professionals to measure multiple parameters simultaneously, streamlining workflows and saving valuable time. Whether you're monitoring pH, conductivity, dissolved oxygen, ORP, salinity, TDS, or using ion-selective electrodes, the HQ 4200 consistently delivers reliable and accurate results to support informed decisions. Its large, full-color display provides clear, detailed data, enhanced usability, and boosting efficiency in analysis. With its rugged IP67 rated design, the HQ4200 is built to perform in even the most challenging conditions. Its intuitive interface includes on-screen, step-by-step guidance for operations, calibration, and troubleshooting, minimizing training requirements while ensuring precision at every step. Designed for versatility and ease of use, the HQ4200 simplifies the complex workflows, making it an essential tool for professionals who demand accuracy and efficiency. Looking for even more capability and efficiency in your water analysis? Upgrade to the HQ4300 and benefit from 3 probe inputs, enabling simultaneous multi-parameter measurements for the most demanding workflows. With the same trusted accuracy, durability, and intuitive operation, the HQ4300 offers enhanced versatility for professionals who need to juggle multiple tests without compromise. The HQ series is a versatile family of water quality meters designed to meet a range of needs. Each model combines exceptional reliability with features tailored for specific applications. From the HQ4200's dual probe input for flexible multi-parameter testing to the HQ4300's 3-probe capability for simultaneous measurements, you can find the perfect solution to match your workflow and achieve accurate, dependable results, whether in the field or in the lab. What's up, Big Steve, with the big $2 Mr. Luxury Hongsloy Premium trademark logo there on the Hongsloy, getting their freak on as we speak on stream. I'm over here just reading the description, Steve. What'd he say? Look, I'm just saying what this, I'm describing the thing that they used to do the test here. Okay? Gosh. Now, I wonder what the calibration process for this is like. But look, I'm getting distracted. This is a rabbit hole that we don't need to go down, right? Right? We don't need to go down. We don't need to go down. Well, they are measuring conductivity, so like maybe we could look at how that works, right? All right, fine. You twisted my arm, bro. Let's open up the manual for this device so we can see how it works so that we can go back and it actually get to reading this thing. So this is the US EPA direct measurement method. Scope and application for brine solutions, produced waters, and hydraulic fracturing waters. Let's see if I'm in the right place. Yeah, conductivity. So we have a beaker, 100 milliliter polypropylene, wash bottle with deionized water, conductivity standard solution. Sample collection and storage. Collect samples in clean glass or plastic bottles. To preserve samples for later analysis, keep the samples at below 6 Celsius for a minimum of 24 hours. Let the sample temperature increase to room temperature before analysis. Rinse the probe with deionized water. Dry the probe with a lint-free cloth. Put the probe in a beaker that contains the solution. Do not let the probe touch the stir bar, bottom, or sides of the container. Remove the air bubbles from under the probe tip. Stir the sample at a slow to moderate rate. If you're doing a field test, put the probe in the sample. Move the probe up and down to remove the bubbles from the electrode. Make sure to put the temperature sensor fully in the sample. Is he reading directions of some scientific equipment he's never gonna buy? Yo, I might actually buy one day, Steve. I might have a windfall. I might have a lottery. I might rob you. All right, you might come to the auction, get too high off your own supply. Oh no, oh no, Steve's wallet fell out. Oh no, I might just steal his whole identity and sell it on the black market. Push read. A progress bar is shown when measurement is stable. The lock icon is shown. Rinse the probe with deionized water. Dry the probe with a lint-free cloth. Okay, now we can go back to the paper. So, they use this probe according to the manufacturer's instructions. The electrical conductivity of all 9 plastic tanks in the 3 groups was measured weekly from 167 days after the plants had grown to 216 days. When the leaves of the plants almost stopped elongating, each group comprised 3 biological replicates. Steve said if they stole my identity, it'd probably make it better. Bro. The pandas are on it too. Just do a 10 to 30% water change and all will spawn. Well, I think the pandas are, it looks like they're definitely spawning. They'll be placing some eggs all over the damn place. I'm sure they'll get eaten expeditiously by the guppies. Okay. For bacterial community analysis, 15 soil samples were collected from the soil, described, blah, blah, blah. All right, let's get to the results of this shit, okay? So I feel like I should have actually, Mick has used Future Sight. It's super effective, yeah. I've been pairing a lot of things with Corey's recently, and I'm just going to say, to all the struggling people looking for an idea. Okay, some of these rare double exclamation mark autos, I am going to place with some regularly spawning Corys because I believe that could be a helpful factor in getting them to get it on. Yeah. Okay, so I kind of was curious what the difference between electrical conductivity is to TDS. And I feel like for me to even read this, I gotta Google electrical conductivity versus TDS. Let's see what Atlas Scientific has to say. They can do this in a... So electric conductivity is closely related to TDS as the dissolved substances in the water contribute to its electrical conductivity. However, they're not the same. Generally, water in its natural state has low conductivity because it does not contain impurities. When water is polluted or has impurities, its conductivity rises. It's important to also note that salt water can register high conductivity levels due to the charged atoms or ions which form when salt dissolves in the water. And Bob said, Nick said the other fish like the watch, or at least they like the smell. Holy. An interesting fact is as water warms, its conductivity increases. For this reason, conductivity is normally recorded at 25 degrees Celsius along with its temperature. Another unit of measurement for conductivity is millisiemens per centimeter, which is equivalent to blah, 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 blah. So they're closely related, but not the same. TDS measures the total amount of dissolved solids in the water. Electrical conductivity measures the ability of those dissolved solids to conduct electricity. In other words, electrical conductivity provides a quantitative measure of the concentration of ions in the water, whereas TDS gives an overall measure of the dissolved solids. TDS meters often estimate TDS by measuring electrical conductivity and implying a conversion factor. This estimation is based on the assumption that all dissolved solids contribute to electrical conductivity. However, this conversion factor may vary depending on the sample being tested and may not be entirely accurate. Interesting. So, which one is better? It's interesting some of the TDS meters use a conversion factor to estimate the TDS, but like they're actually measuring the conductivity. That's kind of interesting. I did not know that. So like, should I just get a fucking electrical conductivity meter and then use that and then apply my own conversion factor BS to it? I don't know. It's kind of interesting though. Okay. We get a little bit more idea of what that is. Now we shall return. What's up, Beto? How's it going? Sutton Aquatics, what's up? Hey everyone, hashtag hardest working. Let's go, man. Well, first it was a joke. Yeah, hardest working fish stream. Not, but we're turning this into a real thing, man. We're owning the title. It's December 29th, New Year around the corner. About to hit this shit hard. Couple fish said probes. Most of the Amazon pins do that. You'd have to have a pin or probe that's specific to TDS. So I think what you're saying is you think most of the Amazon probes do the EC and then have their own little conversion thing. Okay. So they use EC to reflect the water quality. I'm trying to think. I'm trying to think like for fish purposes, is EC or TDS? Do either of them have more benefit than the other? I'm kind of like very curious now. Like the way they describe EC, it makes it feel like because it's like there's more impurities if it's more conductive or whatever. I don't know. There's probably not a meaningful difference. So do people actually look for EC in the water? I got a TDS reader, and it has an EC button, but what it means. Yeah, it is the electrical conductivity, and apparently a lot of them estimate the TDS based on measuring the electrical conductivity and applying a conversion factor thing to it. Excuse me. So they use the EC index to reflect the water quality. The EC index of the different plastic tanks was measured every week from 167 to 216 days after the plants were planted. So they had them planted for 167 days before they started measuring. The average electrical conductivity of the control, which excluded any aquatic plant growth, was approximately 300, whereas the ECs of the group involving the growth of aquatic plants were approximately 150 US/cm, and from 140 to 209 US/cm respectively. I accidentally clicked on the graphic. Get me out of here. Get me out of here. Yo, I'm in hell. Here we go. These data suggest the growth of the aquatic plants significantly decreased electrical conductivity, indicating that V. natans and V. spinulosa purified the aquatic ecosystem where they grew. In addition, the data shows that the V. natans has a slightly greater water purification ability than V. spinulosa, with no significant difference as indicated by the T test. Bravo says, I do know pure water does not conduct electricity. It's the dissolved ions in the water that conduct electricity. So here we see that this is the control where it has no plants. And below here, the conductivity is almost half. It goes a little bit up. No, actually, it does look like it's basically half most of the time for the V natans. The V spinulosa does go up to 200 at the end here, but over here with the control, it's like basically at 300 for most of the time. So it does seem to affect that. Mick says that's why I follow KH plus pH when spawning fish. Cuttlefish said, yeah, but there are non-ionic solids that can dissolve that will read as TDS and not as conductivity. Okay. Now, are those solids like? Is there any reason why they might be considered less problematic than ones that do conduct electricity, and that's why they would just only do conductivity and not check TDS? I wonder how those non-ionic aolids get measured. Turbidity, Bob asks. I don't even know enough to say if that's a good question. Let's see. So for the bacterial community assays, 3,391,282 valid sequences were obtained from 69 samples. Nice. For A diversity of the bacterial community, Shannon index verification curves for sediment and water column samples reach saturation rates, indicating that our data has sufficient sequence covering most of the bacterial community. The SHA-1 index, which represents community richness, ranged from 837 to 3,028 and 211 to 407 in the sediment and water samples, respectively. So it looks like the sediment had a lot more diversity than the water. The SHA index representing community diversity ranged from 8.459 to 10.735 and from 3.707 to 5.569 for the sediment and water samples, respectively. The mean A diversity was much higher in the sediment than that in the water samples, suggesting that richness and diversity are lower in water than in sediment. We found a significant difference between the mud control group and the groups with aquatic plants. In the control groups, one with soil and one with water, the SHA-1 and Shannon indices of CTM were higher than those of BKM, suggesting that water promotes bacterial community richness and diversity. So one was just like mud, and the other one had water, I think, as well. A significant difference in the Shannon index was obtained between the water control group and the water sample, which contained V natans. Together, our data indicate that valsinaria reduced bacterial community richness and diversity in sediment and water columns. Okay. I'm just looking at these graphs and seeing if I can even register them in my brain. These data suggest the largest source of variation in microbial communities is introduced by aquatic plant growth, and that different plants affect the variation in microbial communities. The composition of the bacterial communities was analyzed from phylum to family in all 69 samples. In the mud control, okay, Cuttlefish says, I think CO2 dissolving into water would contribute to TDS, but not conductivity. If used to make carbonic acid, I think it does affect both. Some sugars and alcohols are non-ionic. Okay, that's good to know. In the mud control sample group, Acidobacteriota, Proteobacteria, Plantomycetota, Chloroflexi, and Viromicrobiota were the dominant phyla. Furthermore, our data showed that the abundance of Proteobacteria, Bacteroidota, and Cyanobacteria increased in the VNR and VSR groups when compared with that in the control sediments. Whereas the phyla Acidobacteriota and Chloroflexi were reduced At the class level, gamma proteobacteria and alpha proteobacteria were the dominant classes in the VNR and VSR groups, and the relative abundance increased twofold compared with that in the CTM group. And here are histograms of the bacteria groups at the phylum level. So we got a nice little chunk, not that much cyanobacteria in most of these. In the VSR group, proteobacteria is like, looks like 30%. And over in these two, acetobacteria is, yeah, looks like maybe like a 20-ish% range, but pretty spread all throughout, honestly. Not that I know what that means. Mick says that's the thing. You'll have to learn. You'll have different stats for the free water mass compared to what is possible to measure in the gravel/soil/sand. Yep. The actual way to read 2 TDS is to boil off all H2O and weigh and calculate based on the solids left over. Yeah, that sounds like that's too much work. Yeah, let me stick a pin in it. Let's stick a pin in it. Okay. Let's go to where they're gonna. Jesus, bro, look at this. This is just crazy. I don't know what this is. It looks absolutely nuts, though. But here's some for all you bacteria nerds. I'm sure you're in here, right? Someone knows what this means. This is a lot. It's a lot for my eyes to handle. But it looks maybe that'll make a cool tattoo, you know? So we analyzed the sediment and water. Audio is good, video is no good. What's wrong with the video? Try refreshing. I'm not seeing the normal issues on here on my end. I'm not dropping any frames at all, so it should be good. So blacked out for you? Okay, I would definitely refresh then, because that shouldn't be blacked out. We analyzed the sediment and water column microbial community abundance and structure using high throughput DNA sequencing technologies. Bacterial abundance was much lower in the water columns, as indicated by the A diversity of bacterial community. Okay. The microbial association interactions were stronger in water column than sediment, as shown by network analysis, possibly because water columns act as one unit and the microorganisms interact and communicate freely. While in sediment, microorganism interactions and communication are limited to numerous small units. The growth of V. natans and V. spinulosa reduced the SHA-1 and Shannon indices of sediment and water samples, suggesting that the submerged macrophytes may promote the growth of some microorganisms while inhibiting others. Okay, better, you got it fixed? Okay, good. Thereby reducing bacterial abundance and diversity. The B diversity results of the microbial communities in the sediments and water columns significantly differed between the absence and presence of Thalassaneria species. In the natural wild aquatic ecosystem, submerged macrophytes influence bacterial community composition in their rhizosphere sediments, epiphytic leaves, and surrounding water. Microbial diversity is used as a bioindicator in aquatic ecosystems because it's affected by water quality. Our data revealed differences in microbial diversity and water quality caused by submerged macrophyte growth. The submerged macrophytes improved water quality and affected microbial diversity, reconstructing the microbial community by recruiting specific organisms while inhibiting others. The aquatic plants and interacting microorganisms improve water quality, mutually promoting each other. Proteobacteria, acetobacteria, plantomycetoda bacteria, Viomicrobiota, Bacteriodota, and cyanobacteria were the dominant phyla in the VNR and VSR groups, whereas in the mud control sample group, acetobacteria, proteobacteria, plantomycetoda bacteria, Cloflexi, and Viromicrobiota were the dominant phyla. All right. Some of these increased due to the Thalassaneria sp, aquatic macrophytes, especially for acetoproteobacteria and Y proteobacteria. Similar to a previous study, it has been reported that different submerged macrophytes differ in the shape of their interacting bacterial communities. Our results showed that V. natans and V. spinulosa, highly related V. and highly related Vallisneria species differ in their microbial communities in sediment and water columns. Interesting. So it says, I'll hop back on in a few. I got to drive. I sent everyone I work with home. The storm is getting bad. Oh, damn, man. I hope you're all right. Where are you at? I'm like here checking my weather. Well, if there's going to be a bad storm, no wonder all the quarries are going crazy. Future research using metagenomics and metaculturonomics techniques will provide more solid data to help us use different aquatic plants in the microbial communities to improve and protect aquatic systems. So, basically, according to them, the V-natans of V. spinulosa improve the water quality as the electrical conductivity index decreased. Furthermore, they analyzed the microbial community abundance and the diversity composition of the microbiomes were altered by the Vallisneria, its growth in the sediment and water columns, and the relative abundance of bacteria was higher in the sediment than that in the water columns. The diversity of microbial communities decreased. The A diversity of microbial communities decreased due to submerged macrophytes in both sediments and water columns, and the B diversity also varied significantly with or without the Vallisneria. Very interesting. Very interesting, guys. Yo, no water change, people. Come on. You can spend this, right? Self-cleaning, self-purifying. Like these are some great buzzwords, man. These are some great buzzwords. Yo, Fatherfish, you got to get in on this. Yo, I don't think Louise would mind if I said this, but Louise was telling me the other day. She said that like 2 years ago or something, when Father Fish was going crazy with the resurrection jars, she said that she made so much money off of medicine sales because she had like many customers coming in and telling her they went out, scooped up a bunch of fucking disgusting mud from a local waterway, put it in a jar, and dumped it in their fish tanks. And they thought that they could not feed their fish anymore after doing that because this resurrection jar would provide the food. And they had all sorts of parasites and worms and shit coming out of their fish. And she literally said that like she made a killing on medicine. And like it wasn't like no, people were coming in specifically to be like, hey, I messed up. I went out, I did this, this is what I did, and this is what happened. And she said it was like 20, like we're not talking like 5 people, we're talking like tens and tens of people. It just goes to show you like this is why when people like Father Fish pop up and other idiots, I shit on them so hard because I know it seems like this like a fun content harmless thing, but like people actually listen to these dumb fucks. I mean, it's crazy, but people listen to it. They have a harder time keeping their fish. It affects their life, and I just think that is why talk about not feeding your fish, getting some dirty ass water and and mud and dumping it in your fish tank is a fucking terrible idea. If you're promoting something like that, like bro, I'm gonna clown you until, well, as long as I can, because we don't want to speak ill of the dead, right? Sutton says ice storms are worse than snow. I would do 100 straight days of snow than one ice storm. They're calling for an inch of ice. Damn. Bob said, I didn't know that many people actually did it. Yes, bro. Yes. Yes, people definitely actually did it. It's crazy. But yeah, that like no one likes to be mean. I kind of like being mean, but like when someone's giving out some horrible advice like that, just like straight up horrendous advice, then I'm gonna be mean because bro, you're actively making people's lives worse if they tragically listen to you. Now it is kind of on them for being fooled by a very obvious con artist, but it is what it is. Phew. Pathogens aren't real. Stepfather fish, stepfather fish. Pathogens aren't real. Just wait till stepfather fish realizes that phosphates, nitrates don't exist. It's going to be A bonanza, bro. It's gonna be a bonanza. I don't even know what the definition of bonanza is. If only there was a way I could find out. Hold up. Bonanza. An American Western television series that ran on NBC. Wait, are you for real? No. What's Urban Dictionary say this shit mean? A state of well-being, a state of plenty, derived after famous country slash western cereal in which everything ended jolly and the mood was happy. Oh, you should have come. It was a beach bunny bonanza. Bikinis everywhere you looked. Oh boy. Yo, this is one of those sayings named after some random cultural thing. It's not even a real word. It's named after a TV series that ended happy. Wow. Okay. Bonanza. The more you know. See, this is why I don't. It's googling things. It's like, bro, really? This whole time? All right, we gotta figure out what's up with George Sagittaria though. Like, Val's cool and all, but like, what's up with the George Sagittaria? Because that's kind of now my concern. Like, we got this hybrid disastrous Val, and I even covered the Dwarf Sag yet. It could be so much worse. What if it's worse? What if this one's going to, grow me out of house and home? Denying science can turn on you one day for real. Yeah, it's fine. Whatever. It's a problem. Okay. Give me 1 second. I'm pulling these up. I'm pulling these up. A lot of I keep when I was looking these up, I keep getting like these like galaxy papers of like galaxy. Yo, you know what? We're about to go on like such a. This is about to be such a off ramp, such a detour. It should be short. It should be short. But I found this one. It's from 2021, the Pharma Innovation Journal. Okay, what the hell? Yes, the Pharma Innovation Journal. Art and Science of Aquascaping. The title just I think this is like some kind of school situation. Yeah, man, we got some student department of horticulture. This is out in India. We got some PhDs on here. Okay, okay. Let's see what they define aquascaping as. Not on you, Nick. It was regarding the quote. No, Mick, you're threatening me. Come on, you're threatening me. Deny the twist was gonna be it's not the science I was denying was nature. Okay, you about to pour a resurrection jar down my throat. That's what's about to happen. Okay, aquascaping is the art of arranging and of planting aquatic plants, stones, rocks, or driftwood in an aesthetically pleasing style within an aquarium. In other words, it's landscaping and gardening underwater. Fair enough. Fair enough, Pharma Innovation Journal. An aquascape is an ecosystem. Oh, here we go. Ecosystem. Let's go. No water change for 3 billion years, in which every living and non living item used contributes not only to the aesthetic beauty of the aquarium, but also to the biological and chemical balance that allow the plants and animals to live inside the closed aquatic system. Robert Ringgold on built the first stable aquarium. In his experiment, he used 13-gallon glass container in which he put eelgrass, goldfish, and snails. In the early 1900s, Mr. Takashi Amano pioneered the new aquarium style by then supplying them with lighting, high levels of CO2, and a nutrient-rich substrate that was suited to plant growth. The planted hobby really took off when the Dutch-style aquarium was born in the 1930s. See, Dutch-style men coming in clutch. The Trias worms are beautiful too. I've been using them as a live food for my fry, bro. I'm cultivating detritus worms. Ask about them. Aquascaping styles are constantly developing, and the designs are not only incredibly beautiful to look at, but also a form of intellectual art. combining the imagination, the ideas of the authors, and the ability to create a viable biological system. There are several distinct styles of aquascaping, such as Dutch style, Japanese style, natural style, Iwagumi style, jungle style, biotope style, and paludarium. Of all these styles, Iwakuni style is one of the most popular aquascaping styles today in 2021. Let's take a look at the Dutch style. Linear rows of plants running left to right are referred to as Dutch streets. This style was developed and started in the Netherlands in the 1930s, during which period freshwater aquarium equipment became commercially available. Man, you telling me the Dutch style started this all, man? I'm gonna put the link to this in the chat. I just thought it was interesting. They list all the different styles. They give a picture example, and I thought it was just it was a nice little summary of the different styles of aquascaping. If someone is struggling to write a script for aquascaping video, you could just plagiarize this. So the AI is gonna do it for you anyways. Yo, skeptical, hop on this, buddy. Okay, aquatic plants for aquascaping. What are they recommending here? When we get back to the dwarf sag, but dwarf Sagittaria produce chems. They release to fire their plants by releasing from the roots. That's why they often stay in tanks after a year or so. A scientist told they often stay alone in tanks after a year or so. A scientist told us here. Now my question is that the case if you're doing regular water changes? Because they're doing regular water changes, does that mitigate that effect? And then also I assume that Valsnaria does the same thing, right? But I do know a couple of fish has mentioned that. Why didn't that's why I wasn't rocking with them. So here's what they recommend for plants carpeting 1 to 10 centimeter height. Pygmy chainsaws, Echinodorus tenalis, dwarf hairgrass, Elocaris parvula, Tristerwort, Riccia fluitans, and Java moss. The recommendations for 1 to 20 centimeter height: Anubias nana, Anubias avzeli, Gwenwedinti, Catacomb, Cryptocorn wedinti, Cryptocorn brown wedinti crypt, Undulated crypt, the Cryptocorn undulata, Water barricade, Dipilus deandra. Wait, what is that? What is this? And what if you gravel vac every month? Yeah, that's the question. I don't know if I've Dipilus deandra. Is this a cool plant, a couple fish, or is it bullshit? I can't tell. Kind of looks like it could grow straggly. Hemanthus microthenoides, pearlgrass. Okay, pearlweed. It's an OG plant. Okay. This is 2021, so I mean, I guess things move fast, but it's interesting they add like the common names, probably like the common name you see on like a list. World marsh pennywort, hydrocoil verticillata. Versilata. Bro, I don't know any of these Latin names for plants. This is a cool one though. This is round. I don't see ones this round usually. Now this looks nice. This roundness. Hold up. Yeah, why don't more people have this one around? I know a couple fish gonna be like, yeah, man, yeah, everyone's got this. What are you talking about? But I feel like I haven't noticed this in a billion tanks or anything. Also, what the hell is that red plant in this picture? What the? Yo, what? There's a lot going on here, man. That's a cool one. I'm with them. Old school like black and white pics in the magazine. Well, is it any good or is it BS? I collected hydros at one point. I had 7 var. What do you think? Do you think this is a good one? Sutton Road Aquatics that are you reading a Bible? It does kind of look like that. Nah, man, we're reading the Art and Science of Aquascaping in the Farmer Journal. Baby gashes. What kind of name is that? Microthymum umbrosum. Oh, this is baby tears. Okay. And water cabbage. Ooh, this is the one I want. I want the red one of this. Samulus parviflorus. I do want the water cabbage. I do. I like this plant in theory. Okay, for medium plants, they're saying frogbit, Anubias bardon, Anubias barderi, cophifolia, blixa, okay, bulbitis, all right, cypress, helfin. Maybe they meant helferi. This looks no. This is cypress. Yeah, I guess this is grass-like plant. I'm not into it personally. Porto Alegre sword, Echinodorus, Porto Alegrenensis. What in the world? Whoa, whoa, whoa! What is this? What is this? Wait, this thing kind of looks cool. Hmm. How big does it get? Massive. Yo, Florida Aquatic Nurseries. You don't got a favicon for your Squarespace? This is embarrassing. Come on, at least try. Interesting plant. It's not. It says not a common echinodorus in the hobby, but it is an ovally aquarium plant. Compact round green leaves would make it a suitable choice for mid-ground. Yeah, there's not a lot popping up about this plant. There really is not a lot, which makes me want it. Like this thing kind of looks dope. Now I want this plant. Who's got it? Who's got it? Yeah, this is not popping up for sale. Let's see, buy. Maybe it. Well, let's look up the trade name for it. Is it this Echinodorus red Reuben? Is it a port town? You would know better than me. It's calling the Porto Alegre sword. Let's Google that name and see if wow, actual damn swords are coming up. Aquarium plant. It's crazy. Mm, don't waste money on Brazilian swords. Buy this instead. I got this Brazilian sword plant. This could be it. It could be what's under this Brazilian sword plant name. The peace lily Brazilian sword plant. This one says this Brazilian sword potted plant is called Spathiphyllum tasson. It's also known as the peace lily or Madonna lily. Does this look similar to the pictures of this? Google Porta Alegre. Okay. Yes, city in Brazil. Looks like a very bustling area. I can't really tell if this is the same plant, but maybe. It's kind of interesting. They got broad flame leaf ivy. The town coastal to Uruguay down south. Okay. Yo, this waffle plant. This can't be put underwater, can it? What are they talking about? Brazilian pennywort, golden moneywort, Java fern, dwarf sage, and lizard's tail, Saurus emus. Gosh, bro, they're pulling out some random shit, man. Does this go underwater? Like what? I don't know what. The other pictures are showing it like all the way underwater. They're calling it an aquatic plant. The tall aquatic plants list are just massive. This is kind of crazy. This is kind of crazy. Scarlet sword plant ought to be Echinodorus IMO. Yeah, fair enough. Although, who knows? Maybe when the reason we can't find it, it might have been reclassified. Might have been reclassified. All right, all right. I gotta get to at least one dwarf sag thing before I freaking. I gotta set up this trait. All right. This is gonna be. It's kind of on topic, but a little broader. Now, again, I'm trying to come up with the most recent articles as possible since shit's changing all the time. This is from October 22nd, 2025. I'm pretty happy with that. Wild and globally traded ornamental aquatic plants harbor diverse plant viruses, including notable crop pathogens. Let me see the full PDF real quick. This might be a short one. Well, not that short. Okay, there's photos. There's photos at least in one part. We'll skip around. We'll skip around. Okay, so macrophytes is the trend here. The multicellular photosynthetic organisms inhabiting freshwater and marine environments. Okay. We've already been over how they compose a wide taxonomic diverse group. Some species, like rice, are cultivated for consumption and have considerable economic value. Conversely, several aquatic plants, such as water hyacinth, rank among the most aggressive invasive species worldwide, posing a significant threat to freshwater ecosystems. One of the primary source of invasion is the ornamental plants trade for aquariums and ponds. Several studies have shown that these plants are easily obtained through online. Do you need a study to show that they're easily obtained through online stores and informal marketplaces where sales remain unregulated? But their concern here is what was a little bit more interesting to me. Imported plants can carry microbes, including viruses, which may survive long distances and establish themselves in new environments together with their host. This could result in the emergence of novel pathogens that threaten native populations of wild and cultivated aquatic species. Because recently, some people, like the one thing before, they put an emphasis on sterilization to remove pathogens and stuff for plants. And I think there was the other day, a couple fish and Flash were talking about some kind of disease or something that was causing bumps on the plant. So I've been wanting to look at just plant diseases for aquatic plants in general. I feel like we all are familiar with the concept of diseases for like crop plants because you hear about like these horror stories of like, I don't know, someone's corn getting massacred overnight. But I don't really and haven't thought that much about what that looks like in my aquarium plants. Most aggressive and invasive? Mick said start collecting different plant questions and bring on Kelly Foreman for a stream. She's an expert. That's a good idea. But Mick, you're just deflecting because you're too scared to come and talk to us, bro. What's Mick hiding? Although aquatic plants dominate numerous water-related ecosystems, our knowledge of viruses that affect them remains limited. Okay, we got something in common here. Addressing this gap is important as viruses harbored by these plants can enter nearby water bodies through roots or surfaces running off during rainfall. Let's check out what they did here, man. Let's get past the background. Ooh, mining of aquatic plant transcriptomes for viral sequences. They first searched for viruses associated with aquatic plants by mining publicly available RNA sequencing data from the 1,000 Plant Transcriptomes Initiative. Yo, what's up, Jesse? How's it going? Jesse said, got to get up at 4.5 hours to go get fish, but hey, I'm up. Hey, happy fish day, man. I hope the fish come in well for you. Due to their unavailability at this time, 29 transcriptomes were not considered. Okay. So they're analyzing this shit. Mick said, I'm no doctor or professor, but I'm a nerd. Oh, you fit in perfectly then, bro. You fit in perfectly. The only doctor I know is Bamba. He's got 5 different PhDs in aquascaping. But that's the only doctor that I know. Okay, they're doing all their methods and such. We're not even gonna understand that. Let's get to where they talk about what they found. Trips chromes from all groups of the analyzed aquatic plants contained viral contigs, which were classified into families with exclusively plant host using Megan, MEGAN. They were the most prevalent in angiosperms, where they were detected in 25% of analyzed species. Let's just remind myself what an angiosperm. Flowering plants that bear flowers and fruits. Past Bamba's bedtime, rockabye Bamba, Bob says. That's funny. Okay, that's kind of interesting. Most prevalent in angiosperms, detected in 25% of analyzed species. That kind of makes me feel better. It was only 25%. That was some. Well, let me keep reading. Similar contexts from families associated with diverse hosts, including plants, were most frequently found in angiosperms, 45% of analyzed species, followed by algae, 29% of analyzed species. A substantial fraction of algae transcriptomes contained contigs, with the highest identities to the members of the Phyconaviridae, family, including several that were classified into the genus Phaeovirus. Okay. Here they list all the different viruses that they found, it looks like. Yo, Beat Cryptic Virus 1 goes crazy, man. Look at that. Look at these virus names. Some of these virus names are Sagittaria latifolia. Okay, so this is a plant species they found it on. Bacopa Carolina, they found a couple on. Sagittaria latifolia, they got a couple on. Triglochin maritima, they seem like they found a bunch of different ones on. We got Amaranthus tuberculosis amalgavirus 2. Carrot cryptic virus. Fragaria chiloensis cryptic virus. Rhodiola cryptic virus. Rose cryptic virus. Rose partitive virus. Oh, hey. It's getting absolutely devious. Or as they say, diabolical. Next, they explored the viromes of ornamental aquatic plants through high throughput sequencing, focusing on globally traded species we used for decorative purposes. Okay. Ooh, it's getting late, man. It's getting late where we are just working out. So, we got characteristics of Arigatum yellow vein Sri Lanka virus and its defective DNA discovered in Arthurnia reneki. The colored blocks indicate homologous regions between the two sequences, while the stripe pattern in those regions where parts of the sequences are repeated. So we got a little thing here, some of the codes and what they found. Question is carrying internal and outside of plant surfaces. All right, let's see if we can find that out. This could be infected. Here's another one. Linderina rhodofolia yellow vein virus. Yo, this plant kind of looking cool, man. Look at that vein. That's a vein. Oh, yeah. Look at that. Man, this looks complex as hell. And mostly it does because I just don't know what it means. A couple of fish said inside the cells a lot of the time. Oh, great. Love to hear it. That plant is so weedy, I had to get rid of mine. Does it always have this, like, the vein does look cool, but I mean, that does look like kind of a mess. It does look like, and if it grows super fast with CO2 or whatever, then I could see. Let's read some of these virus names, man. Bean common mosaic virus clade, onion yellow dwarf virus clade, sugar cane mosaic virus clade. The veins are caused by the virus. Okay, that was my question basically. since it was called the yellow vein virus, I had my suspicions. I could do that much deduction. Well, is it possible then that any of these other veined plants that are fancy and stuff like are caused by viruses? And is that a problem? It's not bad, and most are highly special. Okay, see, bro, you're ahead of my question before you even hear it. All of them, all the veins pretty much are caused by a virus. Jesus Christ, dude. Yo, you can't live anymore these days. Not a problem though. All right. Yep. Already forgot it. He said I've been looked into them. I already know. I know. I know you have. You looked at all these damn plants. He's ahead of the game. Looks like the rope fish worked out the differences. Good. Sweet potato feathery mottle virus clade. These virus names are like sometimes the fish names. I'm like, really? These virus names? I'm really raised my eyebrow. I mean, what's going on? Okay. The majority of viral sequences detected in angiosperms in the study originate from double-stranded viruses of the family Colemoviridae. Several partial contigs resembling members of that genera and Kevmovirus and Padnavirus were detected in two aquatic plant transcriptions. Zebra Aquatic says Altherna. God damn it, Reneki have a lot. Yeah, wait, let me put the link to this, by the way. You just gotta go to the download link for the PDF for the full thing, but there you go. That's the link to what I'm reading. I'm feeling weak in the veins just hearing you read about it. Hypochondria is a thing. Seriously. Okay, viruses from Colemoviridae cause some economically important diseases of tropical and subtropical crops. Yeah, so some of the virus names did have like food crop stuff associated with them, so that's one reason why I was kind of getting like, yo, what is going on over here, man? I'm not so sure about this. Pull this back up. Ludwigia, yeah, skeleton is the other one I immediately thought of. Ludwigia skeleton, anything Weissgrund, anything Rosenerberg, the Macrondra variegated, and the striped sword plants. The striped sword plants too. No. Marble Queen sword is viral. I think, man, it's gonna kill all my carrots, man. It's gonna kill all my carrots. While some can integrate into the host genome, resulting in the formation of endogenous viral elements, some sequences identified here may also represent EVEs, the endogenous viral elements. Steve said, Jesse, that prawn is doing good, and Bob is literally about to eat paint and saying, now I want to lick those plants. Jeez, what's wrong with you, Paul? We also discovered several partial contexts related to the members of the Rhabdoviridae family in the transcriptomes of Sagittaria latifolia and Murophyllum aquaticum, as well as in sequencing the library of Murophyllum sp. Guyana. This discovery reflects a broad host range of members of the Rhabdoviridae family, which are known to infect various monocot and dicot families. Viruses from this family have been previously identified in the transcriptomes of several decorative plants, including Bacopa moniri, Nymphia alba, and the lace plant Aponogean madagascarensis, as well as in the seagrass Zostera marina. Yeah, the Bacopa seem to have a decent amount. The prawns cool as shit to watch. Yeah, those prawns have been tempting me. I just already have, those giant pink crayfish. The female has a bunch of eggs. And first of all, I really need a real tank to put them in because they've gotten huge. But I think I'll wait till they release the babies. But second of all, bro, what am I gonna do with all these crayfish, man? They got so big. Steve said, I know I heard my cousin talking about mosaic disease and figs too. Oh, fantastic. Furthermore, we identified several sequences with the highest similarity to members of Artitiviridae family, okay? Partidaviruses are characterized by their persistent lifestyle, asymptomatic infections, and their inability to transmit horizontally. A similar trend was observed in the only other comprehensive study of wild aquatic plant viromes performed to date, where cryptic viruses from the family Partidiviridae were detected in 7 aquatic plant species. Out of these, 5 belong to the same order, Alistmatales, as Trigloshyn maritima and Posidonia australis. And Mick said, thank you for sharing a couple of fish. Mosaics and breaching viruses are found in every plant family I know of. Most research is on tobacco, squash, cucumbers, melons, and other agriculturally significant plants. I was trying to slur my words like I just had my like 5th glass of vodka. Okay, while the majority of viral sequences detected in this study represent putative novel virus species, as we've seen with other HTS-based studies, we also identified several known crop viruses in different species. We detected lettuce chloris virus, known to affect crops such as lettuce and sugar beet, as well as beans and tomato. In the transcriptome of M. aquaticum, the myrofilm aquaticum, and in the sequence library of ornamental aquatic plant Reneki rosenervig, and in other decorative plant Lobely cardinalis mini, we discovered the complete genome of cucumber mosaic virus. Come on, man. Zebra Quack says we have a farm by my house with a field filled with mosaic virus. It's creepy. Oh, fantastic. Plants and minerals. A couple is that guy. Get you a man that can do both. He can give you asbestos and a very rare and exotic plant virus. A couple of fish at Hydro Sunset also. Oh, damn, I'm screwed. I got that all over. This virus is known to infect an extensive range of plant species, including crops and wild plants. This observation supports prior findings that cucumber mosaic virus can infect several species from the genus Lobelia, including ornamental hybrids. Other notable examples detected across transcriptomes of various species include turnip yellows virus, Schlumberger virus X, and Cymbidium mosaic virus, each known to affect a variety of economically important plants. Our findings indicate that aquatic plants could play a role as reservoirs for viruses infecting crops, especially given their ecological role in broad distribution. We hypothesize that aquatic plants may link aquatic and terrestrial environments by acquiring viral particles from the water, which can then be spread to nearby vegetation by vectors feeding on their emergent parts. The potential of aquatic plants that harbor possibly pathogenic viruses, including such that infect crops, raises additional concerns about the unregulated trade of ornamental aquatic plants. The commercial trade of these plants is known to be a major source of invasive species introduction, threatening various freshwater and marine ecosystems. Although the sale of some exotic plants is banned in many countries, studies have shown that they are often easily attainable through online retailers or informal marketplaces. Such trade not only introduces invasive species, but also facilitates the spread of their microbes, which may spill over to the native hosts. We detected numerous viruses in the sequencing of ornamental aquatic plants. However, for the illustration, the risks associated with their trade and possible introduction to new ecosystems are discussed in depth. For novel begomoviruses and novel potiviruses discovered in the study. Begomoviruses are quarantine pathogens in the European Union, the UK, and Switzerland. Both A. reneki and L. rotundifolia, harboring begomoviral sequences, displayed ornamental patterns of pink or yellow veins, a desirable trait that can enhance their sale. The absence of these patterns in different reneki variety mini, in which no such viruses were detected, suggests that the vein yellowing could be caused by a viral infection. This hypothesis is further supported by the episomal nature of detected begomoviruses, indicating the active replication in the host plant. The appealing visual traits associated with the virus-infected aquatic plants may lead to their selection by growers, potentially contributing to the faster spread of these viruses through the global trade. While the detection of these begomoviral sequences is concerning, the adaptability of these plants to new environments should be considered when evaluating the associated risks. Okay, I get it. So they're like, yeah, they got these viruses, but they can't survive that well in certain environments, then like maybe it's not that big of a risk. for some of them if they can only survive in warmer regions. Yeah, the bigamovirus sequences were detected in plants native to warmer regions, which could, to some extent, limit their establishment in colder climates. However, the ability of many aquatic plant species to adapt to various environments suggests they may also thrive in temperate habitats. Alien aquatic plants have been shown to survive in colder climates and ecological niches, such as thermal streams, with water lettuce being a key example. If a virus affected alien aquatic plant were to be established in ecosystems, Vic said a pistol male looking for Cory eggs. Yeah, yeah. This tank has basically only had things in it that love to eat panda Cory eggs. But the entertaining thing about watching it is sometimes the fish seem to look as hard for the panda Cory eggs as I do. So good luck to him. Hey, maybe it'll help him encourage his spawning. Okay, get your nutrition up, man. Now I know it looked like he was luring the female to a cave, but the female has definitely been more reclusive. So, oh my god, look at this. So I need to catch this guy actually, but this is one of my Beta Alpha Marginata I raised up, and he was just like thrashing around to get the. And I had them. This was gonna be a Beta and Cory tank, but the betas were just so brutal. They were so quick and smart to eating these panda Cory eggs, as you can see, that it was just like I know the pistol's looking, but just imagine more than one of these betas in here. They're really on top of it. And that looks like a pretty ass. There might be a male going out there. It looks very nice. The betas are one of those ones that I want to give a display tank, a proper display tank to soon. But yeah, that beta is intense, bro. Look at them. They both want the damn eggs. They both want the damn eggs. Beta's gonna get more of them though. I'm telling you. They also detected novel potyvirus, Sagittaria virus A, in several ornamental plants. Okay. Let's see what they conclude here. With all these words, their conclusion is 4 bullet points. You gotta love it. You gotta love it. The analysis of publicly available and newly generated HGS data has expanded the understanding of viruses associated with aquatic plants. Both known and novel viruses from 2 of the largest plant viral families, Potyviridae and Gymnoviridae, were identified along with viruses from several other economically important families, including major crop pathogens. The detected viral sequences represent a variety of genome types reflecting the disease viral populations in aquatic plants. Viral sequences were found across all investigated types. Like I said yesterday, a community tank is not a breeding tank. A community tank could be a breeding tank. I get some panda cores out of here. A community tank is not a I'm trying to breed wholesale quantities tank. But I mean, breeding happens in them. Fish survive here and there. A community tank is not a breeding for profit tank. But you could breed stuff in community tanks. It's not going to be optimized. Now this one is a particularly challenging one. Some things, you can have they do lay eggs in the mop. Nick said, you know what I mean? I do. They do lay eggs in the mop. Now this mop is a little bit thin, honestly. Like I would need a thicker mop anyway. But like with some killifish, like where they're gonna lay the eggs. You have a certain amount of time for all the eggs are eaten. You got thick enough mops, and we're gonna survive. The pandas, like sometimes they lay the eggs deep. Sometimes it's not so deep. Sometimes it's in moss. Sometimes it's on the glass. Sometimes it's on. But when there is nothing else in there eating the eggs over time, the mop does accumulate a nice amount of panda Cory eggs. So I do think it's a mob spawning for the panda Corys. I think is probably a pretty decent strat. Viral sequences were found across all investigated types of plants. Okay. The discovery of potentially pathogenic viruses, including regulated species, underscores the risk associated with the global trade. All right. To fully assess, further research is needed. I hate reading that in these damn papers, but I get it. Viral infections may induce ornamental phenotypic changes that are selectively favored in globally traded aquatic plants, which is really aiding English spread. I do think that is interesting, and again, since I don't understand that much about aquatic plant pathogens and how they like, it doesn't literally. I don't know all these diseases well enough. I'm like, oh my god, fuzzy carrot potato virus. That's in that plant. That could ruin me. That could eat me out of house and home. Like, it doesn't really register for me, but just it is kind of funny that the virus, like some of these viruses, are why the plants are being traded in the first place. Like, they make the plants look nicer. So like we're selecting for the virus, most virus filled, most disgusting, transmissible, pathogenic plants possible. Now, a couple of fish did say he looked into these, and most of them are not a problem and highly specialized and all that. So I'm not personally worried about it for my own plant pathogenic safety, I guess. But it does definitely seem like something worth researching. All right. Now, I would just end the stream. I would. The problem is, I have not yet talked about dwarf Sag. Evolution and progress never stops. Indeed. Indeed. Ooh, you know what's a cool one? We gotta look through this another time. This is just like interesting. Come on, why is it not working? Okay, the link is not working to this damn thing. I'm gonna just put the link to this in the chat. I'm not gonna go through it because it's not about George Sag, but I thought it looked kind of interesting. Abstracts, contributed papers, poster and oral, workshops, colloquia, 94th Annual International Conference of American Society for Horticultural Sciences. It's just a bunch of abstracts in like one long list. I thought it was just kind of interesting. So I put the link to that in the chat, but when I copy and paste the link, it's not working. So I don't really know what's going on with that. So that was totally useless for me to bring up. But maybe if you Google the title, you can find it. Okay, where's my dwarf Sag thing? Hold on. One moment, please. It's giving me a verify. Click all the pictures that apply situation. So I just got to get through that. Here we go. Dwarf Sag paper. It's finally here. The last item in the title that. Oh wait, I talked about Reddit too. Jesus Christ! All right, strap in. We gotta work through. We gotta work through everything in the title. I'm gonna be sued. Yo, look at look at the beta. The fucking Apisto fighting over who's gonna get to eat the panda cory eggs. This is ridiculous. I will get a panda cory, dedicated panda cory breeding tank eventually, but I've just kind of enjoyed having a tank of just a bunch of panda cories all over the place. Like, you see that this is a fun ass tank to watch, and I'm slowly working on it looking better so it can be something that I'm more proud of. But this tank is a good example. When I say I want to be a better fish keeper next year, this tank is kind of an example of what I mean. I enjoy watching this tank a lot. It's an active environment. From what I could tell, the fish in here seem to be relatively healthy. And when you think about a community tank, like once I finish planting things up, hiding some filters and stuff, when you think about like a community tank, like it's like, okay, you got the cories, you got the apisto, you got some guppies, and they all kind of look nice, and they're all playing nice. I got some killies in there. It's just like a fun community tank that looks nice and everything's, fun and et cetera, et cetera. And to me, this is what I want to achieve more of next year. Like Nick said, revealing natural behaviors. It's awesome. Because a lot of breeding stuff is watching fish. There's a lot of fish. I'm not in a rush to breed. I buy it, I watch it for a long time, but sometimes watching how it interacts with other things, even if it's not expressly from the area they're from, can help you understand why they lay eggs the way that they do. Like, what do they need and what they need for eggs being laid to be successfully hidden? Because there are some ways for some fish I found to community breed them, like the scarlet batis and the Elsoma gerberti. I've been able to produce pretty decent amounts in community tanks, and it's mostly moss and rock piles. You know what I mean? Now, those are kind of easier because those are both fish that express parental care, where the males guard the eggs. So then all you really need to do is provide an area near the where the eggs are laid, where the very, very small fry can get to. Because if you think about it, Elisoma and Scarlet Batis are very similar in a lot of ways. They're both small micro predators. They both guard the egg location, and they both have really, 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 really small fry. So you get the moss, they or whatever, they lay the eggs on it. I think this Batis can be like cave spawners too. So little rock crevices and stuff, they might just lay the eggs right in there. And then once they hatch, you got the rock piles and stuff nearby. And then, bam, the fry are in there. Well, if I got any of the fish that I'm looking at right now, they're not going to fit in those rock piles. Fry are going to hang out. They're going to eat stuff. They're going to come out when it's dark. They're super small and hard to see anyway, and they're pretty fast. They dart back into the rocks. And even though it's a take with small volume and stuff, like there's some fish like that you can set it up to where the babies will probably make it. Now, also, I keep a lot of smaller fish that aren't extremely predatory, which helps. But you know, the mouth brooding bettas, that's a great one. They don't seem to really eat their fry that much. The fry come out really big, so that the list of fish I have that could eat the betta rubra fry that come out at the size they do is already limited just off the bat because they come out a bit bigger and they're relatively conniving, so they're not gonna not all fry are equal in survival ability as well. Um, so yeah, there's some things you can little interactions you can come up with. Like I think I'd make a thicker if I made a thicker mop, right? How I would try and tackle this problem next if I were gonna try and up the yield of my eggs in this tank. This mop is super thin, so it's not hiding anything. The cories do really vigorously work them way into the mop. If it was thicker and they're going super far into the mop and they were injecting the eggs deep enough into the mop, it basically be like how I'm doing with the porous sponge mops with the Procatopus and stuff, right? The Procatopus shoot the eggs so deep into the sponges, none of the other fish in the tank with them can even get to the eggs to eat them. So I can pick the eggs whenever, that's fine. The panda cories do actually put their eggs relatively deep into things sometimes, to where I do think it would be challenging for those eggs to be eaten. So I think, and actually, I have had a thicker mop in here, and I was able to get eggs out of it pretty regularly and had stuff. So I think if I get a thicker mop in here, round it really tight, maybe have a tight bunch somewhere, I'd be able to get a decent amount of eggs out of there. I think so. That's how I would try and troubleshoot that problem. All right, I'm getting so like distracted. Okay, so I have to just technically hit everything in the title for at least a second. It's the letter of the law, not the spirit of the law. Like, what's going on? I believe pandas in nature don't spawn in this bright light. Yeah. Well, the lights are supposed to be off too, so you're probably right about that, bro. The PDF button keeps like disappearing. Are you seeing this? Download PDF. Oh my gosh, bro! Wow, all the dwarf Sag papers, like they're trying to paywall them, bro. What are they trying to hide? The dwarf Sag agenda? This is actually ridiculous. So check this out. Biology and management of Sagittaria latifolia, wild, broadleaf arrowhead for wetland restoration and creation. I'm going for this one because I'm trying for applications in our tank, right? You talked about how they release the chemicals that kill the plants. I'm kind of curious if that's going to come up in any of these because they seem to be really enjoying using these plants as a way to restore water quality. This is a totally different plant than fucking what I was thinking of, I think. It's kind of hard to tell from these photos. They love to just do the flower photos and everything. All right. This is a big ass fucking plant. This is not what I was necessarily thinking of. Holy, the water plantain. I think actually does kind of look cool. This could be cool to do out of like a big tank. You know what I mean? You get these big ass stalks coming out. You need a huge tank. 125. Damn, man. I can't use it. Okay, I'm posting like this paper in the chat, but this is not what I thought it. I got my things mixed up here. Got my things mixed up here. It's so hard when you don't know what you're doing. Hold up. Hold up. Mm-mm. There's a lot on this dwarf arrow leaf situation. I kind of want to just shut up, no longer say anything on stream, and just stream the Panda Quarry tank for a while. And just watch it myself, mute up. I might end the stream, as in like I'll stop talking. I might just leave the Panda Quarries on for like a couple minutes. But I do kind of want to throw a thicker mop in there. Kind of interesting. I wonder if I have any mops that are decommissioned I can put in there. Okay, give me 1 second. I'm so close. I am so close to getting the paper up that I was looking for. Because if I don't talk about this, then I'm a liar. All right, here we go. Link to this in the chat. The growth of Sagittarius subulata in interaction with hydrilla. Now, I think we were looking at hydrilla earlier. This is that's that super invasive plant here that kind of looks like an anacris situation. And I'm hoping that this might cover some of the biochemical interactions that have been mentioned. So let's go. Let's go. Let's go. Maybe we'll come through. Let's find out. Let's check out the intro. An aquatic plant interacts with members of its own species as well as individuals with other species as it colonizes and grows in the area and competes for water, nutrients, and light. This interaction may be separated into categories of allelopathy and competition. Biochemical interactions which can result in inhibitory or stimulatory effect of one plant over the other are referred to allelopathy. 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 This is the chemical warfare we've been speculating on. While competition is generally reserved for those processes of one plant that result in the reduction or removal of a factor required by another plant for growth, it is well known that hydrilla will colonize and dominate a body of water by excluding growth of other aquatic plants, primarily through competitive effects such as reducing nutrients and light for other submerged plants. For example, hydrilla has the ability to photosynthesize light conditions lower than that required by most other submerged plants. Hydrilla can utilize carbon dioxide early in the morning when the supply is high, and by the time the other plants receive enough light for their photosynthesis, the supply of available CO2 has been reduced. Another competitive advantage exhibited by hydrilla is its habit of branching profusely near the surface, forming a dense mat that blocks light penetration to plants growing below. Certain spike-rush plants, such as dwarf spike-rush, Elocaris coledoensis, and slender spike-rush, Elocaris acularis, have shown allelopathic potential. So that's that chemical warfare potential. These spike rushes have been found to replace uh, Eladia cadenensis, American pondweed, and sago pondweed through non-competitive effects. This allelopathic response with slender spike rush may be due to the compounds with molecular weights between 600 and 1,000. Established trends of the native Florida aquatic plant slender arrowhead were found to be more effective than the spike rush in resisting colonization of sprouted hydrilla tubers. When hydrilla was grown for 8 weeks in established stands of slender arrowhead, its shoots its shoots, dry weight, and tuber production were reduced to as much as 95% and 90%, respectively, as compared to hydrilla cultured alone. It is not known whether allelopathy or competition was the primary factor responsible for reduction of hydrilla growth. So, in addition to provide additional information on the interaction of aquatic plants, a study was conducted by allowing the Florida native arrowhead plant S. subulata, hereinafter referred to as arrowhead, to develop established strands. So, got the Sagittarius subulata. The last one was like the different arrowhead, so just double checking that this is the damn right dwarf sag plant I'm looking for, not some monster. Okay, just double check. So let's see what they did here. Experiments were conducted outdoors in concrete tanks at the Fort Lauderdale Research and Education Center. They give the exact coordinates. The dimensions of the concrete tanks were 3.1 meter in width by 6.1 meter in length by 0.8 meter in height. The tanks were supplied with flowing pond water. Water level in the tank being maintained at a height of 0.31 meters. Flow was sufficient for 3 changes of water every 24 hours. Pond water was from the same source as described by Stewart, 1984. Well, thanks. I'm just totally, I know exactly what's going on. Water temperatures were measured with a U-shaped maximum/minimum thermometer placed 15 centimeters below the surface of the water. Readings were collected from the thermometer 5 days a week, generally between 3 and 4 p.m. Temperatures of the water were calculated as a mean of the minimum and maximum values measured for the growth period of each experiment. Fertilizers used for this study consisted of Osmocote 18-6-12, formulated with an 8 to 9 month release rate at a temperature of 21 degrees Celsius, is Migran and Dolomite. Arrowhead plants used in the study were from populations of plants maintained in sand, amended with controlled release fertilizers and outdoor culture tanks at the Fort Lauderdale Center. Sprouted hydrilla tubers were obtained as described by Sutton in 1986. Culture in the sand amended with controlled release fertilizer. This portion of the study consisted of planting arrowhead plants in plastic culture pans with dimensions of 27 centimeters in width by 33 centimeters in length by 14 centimeters in height, filled with 15 kilograms of air-dried quartz sand emitted with fertilizers listed in Table 1. Each container was planted with 5 arrowhead plants, and each nutrient treatment was replicated with the 4 pans. The pans, a total of 24, were placed in 4 rows in the tank, parallel to the flow of water, with the 6 treatments randomized within each row. The arrowhead plants were planted May 27, 1986. We're reading about some plants planted before I was born. Holy. And harvested on January 7, 1987. Periodically, water in the tank was drained overnight to help control growth of algae. Okay. And they go on and on and on about the methods. Here we have the fertilizer amounts. We got different amounts of osmocote, isbergran, and dolomite. The highest of osmocote they did was 35. This is in grams, 35 grams. 0.9 grams of isbergran was the highest, and dolomite was 5.6 grams was the highest they had. So let's look at the results. Water temperature averaged 29 degrees Celsius with a minimum of 16.5 degrees Celsius and a maximum of 46.5 degrees Celsius for the arrowhead plants cultured during this period. Under natural culture conditions, it's not uncommon for this plant to be exposed to drying conditions during low tides. Therefore, it's assumed that the periodic drying of the arrowhead plants to control algae growth did not interfere with their growth. Dry weights of the arrowhead plants cultured in the sand amended with the various levels of fertilizers are shown in Figure 1. Okay. The data suggests that a longer culture period, additional fertilizer, or other unknown factors would be required to achieve similar growth of arrowhead plants under semi-controlled conditions at the Fort Lauderdale facility. I'm looking for the conclusion. I feel like we're missing a page or something. Oh my god. No, we're not missing a page. Let me see. Try weights of arrowhead plants. Here we go. Shoot weight of hydrilla plants grown in pans without arrowhead plants were 1.8 times heavier than shoots of hydrilla plants in the established strands arrowhead plants. However, no difference in root weight was observed. The number of tubers per container was 59% lower for hydrilla planted in the established stands of arrowhead plants as compared to hydrilla cultured alone. Also, dry weight of the tubers for the hydrilla plants grown with the arrowhead plants was 3 times lower than for the hydrilla alone. The average tuber weight was 20% lower for those collected from hydrilla cultured with the arrowhead plants as compared to tubers from hydrilla plants cultured alone. It's not unusual to find population of arrowhead occurring as small monocultures. So this is like what a couple of fish were saying, how long term they end up being the only thing in the tank, right? It's not unusual to find populations of arrowhead occurring as small monocultures in various bodies of water in Florida. Even though the arrowhead plants exhibited a slow growth rate as compared to hydrilla and a low biomass as compared to plants collected from field population, this study does show that this submersed arrowhead plant will resist the initial colonization by hydrilla. Long-term growth studies will be required to determine the nature of the interaction. This was like almost a year long. They want to do it even longer, but kind of an interesting older paper. And it looks like, man, I need to get this dwarf sage out of the tanks with plants I care about, man. Oh shit, the piano battery just went out. Let me replace it. Ugh. Yo, here we go. All right. There's a little bit about some dwarf sage. Now let me pull up some Reddit thing. Then I'll fulfill my contractual obligations to you degenerates. And again, shout out to everyone that's here, but also shout out to the replay crew. Big shout out to Cam Jam. Thank you, ma'am, because they are the example of how I like people to leave comments. For the last stream, they just left a comment at every point in time in the stream where they had some kind of something to say. And I like that because then even if you missed out on the chat, like every time you have something to say, you just comment at that point in the video, and later I see it and I can respond back. And everyone else that comes to the video also and watches along can comment as they go. And I don't know, it's just nice, you know. If you miss out in the chat, you feel free to leave like 20 comments as you're watching through. I think it's kind of fun to see the reaction at different points in time as you're watching. All right. Fiscal Pay says Dwarf Sack and Val have been the easiest for me. Yeah, they're really nice to carpet and stuff. But maybe if you're having issues with some other plants and takes with them, chemical warfare may be occurring. I do kind of wonder if it's just something that they release if there's stuff above them. You know, I've definitely had some tanks where floating plants didn't do well. And maybe if you're having issues with floating plants, you should see if you've got dwarf sag in there. Maybe that could be contributing to the problem. Just a thought. Just a thought. Okay, here we go. I gotta pull up a Reddit thing I think you guys will enjoy. Okay. We mentioned the betta fish being white elephant gifts. Now, this was on a stream that is now legendary. It's currently privated, you know? It's just such scandalous copyright material that I need to just hit a button and it'll edit it out for me. But I just, for some reason, haven't done that. But on that stream, we talked about how there was a trend of betta fish being given away as white elephant gifts. Fish Cafe said, "Tiger fowl as tall as me. I'm 5'6". Damn, bro. You know, as tall as I am, man. That's some tall ass fowl. So I gotta see what you guys think of this. This to me was kind of wild. Okay. So this Reddit post here, the title of it is, "What do you do with your deceased fish?" Okay. Now, I would just want you all to ask yourselves, man, what do I do? What do I do with my deceased fish? Okay. And then I just want you to see if it lines up with some of the things going on here. So this person lost their betta. I don't know what to do. First of all, damn, this betta does look jacked up even in this photo. Okay. Okay, it looks better in this photo. I live in an apartment complex. Burying her seems a little silly. Flushing her seems wrong and disrespectful. What do you guys do with fish that have passed? Now, the top most upvoted comment is I bury mine in flower pots. Okay. This person here also burying. This person buries them in a potted plant on the balcony. This person goes out in nature and finds a nice spot to lay them to rest. Okay, I know everyone worried about spreading parasites and pathogens are just like going crazy. This person did pop in to say flushing dead fish is bad practice. If a fish died of bad diseases, it could contaminate local waterways and potentially hurt local wildlife. Yep. I have buried fish myself before. These days, I've kind of been like sealing them in bags and putting them in the trash. I buried them and flushed. I lean towards bury these days, especially the big one. The bigger ones are where it's more of like, what do I do? Mix. I had tanks with giant valve well over 7 foot. Jeez, big ass tanks. This person says water from toilet doesn't go to waterways. It's not the same as store water. It goes to a waster facility with poop, pee, blah blah, and it's clean, sanitized. Okay, so this person says maybe it's not a problem. Cuttlefish says if it's a rare species, I freeze them in a container just in case I want to mount or preserve them. Okay, well we're gonna talk about you in a second. Don't worry. But overwhelmingly, a lot of people is burying the fish in their plant pots on their balcony. That was seemed to be an overwhelming thing. It seemed to be quite an overwhelming thing. Again, house plants is not allowed to flush them down the toilet. I would have to throw them in a residential waste bin. I do sometimes, but normally I freeze them for a friend who likes to dissect them. Okay. Okay, this person puts them in the bin. All right, flushing's a terrible idea. Snails get to them first. Just buried one of mine in a houseplant yesterday. SBC said I feed mine to my terrestrial ice spots. That is what I now currently do as well. Or my mealworm culture I feed them to. I avoid giant valve for that reason, Mickham, but my tiger valve is still ridiculous. Next talk about is 17, 7 foot. Okay, this person, this is what I said. We're talking about a couple of fish here. Now, this was terrible. I'm sorry. I I think this is just disgusting. Do the isopods clean the bones well? Yeah, I think so. Especially the like I have no a lot of them got springtails on there too. So yeah, isopods, springtails. One of them's got isopods, springtails, mealworms. Do they disarticulate them? Well, now you're using words I don't know the meaning of, so I don't know how to answer that. But I'll be honest, I haven't really paid that much attention to it, and the bones do tend to get buried in the core pretty fast. But I have seen like skeletons previously. So this person, they are trying to preserve them, and they followed this video, aquarium fish taxidermy. Okay, I'll put the link to this video in the chat. You guys, I'm not gonna watch this whole thing here. You guys can evaluate what this method is and if it's legit or not. I'll be honest, I'm not so sure about this one. But SVC said his isopods eat the bones too. Well, are you feeding them to the dairy cows, man? Not all isopods. Not all isopods. No, I mean like preserve them in stains and jars as wet specimens. Well, okay. I know what I had a shark that was preserved like that, a baby shark. This is fucking terrifying. If you do this, get the bro. What kind of serial killer ass shelf is this? We got what look. I don't know if these are 3D printed. We got some shitty ass cats, all looking the same, all looking creepy as hell. Okay. Then underneath it, we got this satanic shrine to bro. They did not. This is supposed to be taxidermy, bro. This looks like some fish jerky. Okay, if you're gonna preserve me and I look like this, don't even bother. Yeah, throw me in the landfill, dog. Yo, look at this. Come on. Don't put this. You put this on display. This looks terrible. This looks horrifying. Your house guests are going to be running for cover. They're going to be in the bathroom hyperventilating. I go to someone's house and I see this combination of these cats and these fish. I'm going into the bathroom. I'm hitting the blue light button. I'm rubbing my face on the left side, tugging my right ear. I'm going up like any sign for help that I could possibly do to preserve my safety in this situation, because I may have just wandered into the den of a cold-blooded killer. Okay, my God, bro, please don't do this. Please don't do this. Make them into lamps. It's horrifying. I can't look away. Yeah, this is bad. And then over here, they just got a dead bee in a jar for some reason. Okay, the aesthetic of this, and look, there's empty space under here. This is where you're gonna go. Okay, you come over to eat, they're gonna be chopping your body parts off and putting them on this blank space. There's plenty of space, man. Your toe could be right here. Okay, I don't know what's going on over here, but the aesthetic, the vibe, everything about this assemblement is tragic. Absolutely tragic. Bob said serial killer starter pack. Yeah, what Couple of Fish is saying they're talking about that makes more sense. Preserving them in a jar or something like that. Yes. So it actually looks like it used to be. But this is interior design malpractice. Okay, he should be sued. He should be fired, license revoked, everything, everything. Now there is a more disturbing. There's a more disturbing answer that I'm looking for in here, and I don't know. I wonder if did it get deleted or something? Because where the hell did it go? Let me see if I got a screenshot of it. because sometimes I do these kind of things. All right, here we go. Now, this is crazy. This actually had upvotes when I saw it. Now it's barely in the negative. But this person just says, feed to dog. You feeding your dead fish to the dog, man? Is this what we're doing? Is this what we are doing? Then someone comments before. This got 4 upvotes. I thought I was the only one. Yo, what kind of like coming out is this? You're in the comment section coming out as someone that feeds fish to their dead fish to their dog. Yo, yo, seek help. But sometimes water changes get my fish. It could be the same water change that they've been doing for years now, and someone just sends it to it, and what does it make it? No telling if the stage is putting too much chlorine at that point or what happened. I do use prime or safe, but fish are sensitive. If this is happening that often, I have questions. What do you mean a couple fish? I mean like using this method. Or sometimes fish just die. So my dog has gotten a particularly large severums before and multiple large imbunas. I've only buried 1 fish, and that was a large green severin I started with years ago and passed away recently. I didn't think I was going to be that upset over it, but I was. Bro, are you guys feeding your your dead severums and imbunas to your dogs? Like, let's talk. Like, do we love the dogs or not? Do we love our dogs or not? Because like, if I love someone, I'm not feeding them a fish that died of a mysterious illness, reached a untimely demise, and who knows, it could be dead for unspecified amount of time. You're gonna take that and feed it to your fucking best friend, man's best friend, the dog. That's disgusting. That's disgusting. I'm just like trying to, the mental image of a dog is chomping down on some raw, half dead, smelly, large severum. He said a particularly large severums. Several particularly large severums. It's kind of a disgusting mental image. I'm not gonna lie. Now, maybe someone in that chat is like, "Yo, I've been doing this all the time, bro. You're bugging." But that's why I'm asking. Like, is this what we're doing? This is what we're doing. Fiscal Bay said dogs are disgusting. The grosser, the better. I mean, I do think like I'm kind of biased. I'm team cats. I like cats, bro. I like cats. I do think dogs are they're a little bit gross, man. A little bit gross. They lick me a little bit too much. Pause. Just a little bit. Now, dogs eat their own poop. What's up, Potor? I've been fishing too much to appreciate fish intestines. You're not feeding dead fish to your dogs? Well, dead aquarium fish specifically to the dogs? That's not you? What's the problem, Nick? Come on. Live a little. I'm Team Cat too, but I groom dogs for a living. Oh, that's cool. One of my LFSs, they own the LFS, but then next door they own a dog grooming business too. It's kind of interesting. So there's definitely a lot of crossover with some just pets in general. If you got pets in general, you're more likely to have these damn fish. That seems to be the case. Now, I don't think I went over this on stream before, but I thought it was kind of interesting. Shrimp Up Aquatics. Has anyone here ordered from Shrimp Up Aquatics? I'm just like a judgmental asshole. The name Shrimp Up Aquatics, it just sounds like they're marketing to idiots already. That's like my assumption. This is like one of those To me, this is like one of those Facebook companies I only run into in Facebook comments. You know what I mean? This is crazy though. Yo, this gotta be a red flag. Free shipping for orders over $30. God damn, bro. $30? That's it? Yo, they're shipping this to you ground. This gotta be the ground shipping, bro. No way. No way. Free shipping about $30. This gotta be some garbage can shrimp. Let's see what they got going on. Yo, so West Say shrimp. Oh man. Oh man. They got Suese. Well, it's sold out, but they're selling them for 20 each. Okay, we got the Skittles shrimp, 5 shrimp for 17.49. I always hate these. Like rinsing fish is a nasty job. Yeah, we got fire red cherry shrimp, 1 shrimp for 3.50. Blue Dream for 3.95. I hate all these. Just round it up, man. Just round this shit up. Blue Velvet for 3.99. Okay, so we got like $4 shrimp, green relief for 8 each, basically. Snowballs, 4th of July shrimp pack. Now, I do think more people need to do shit like this. It actually does work. It is incredible. Look, red, white, blue. Like this shit sells. People love theme things. Halloween colored stuff, goes Halloween, like all these different seasons, like ornamental, man, ornamental. We got crystal red shrimp, 1 shrimp for $7. We got a mono shrimp from $5. Shadow pandas at 10 each. Golden bee shrimp. I like golden bee shrimp. I need to get some of these in 2026. I like the jibe with their jib, whatever. Pause. I don't know. Red Galaxy Pinto, 25 each. Holy. Crystal white shrimp. Oh, these are kind of funky. All right. We got the pom poms. $20 a crab. Christ on a stick, man. It's crazy. Vampire crabs, 25 each. All right. Let's see that. See that. But shrimp aquatic. So this person is complaining. I did make comments in this thread. I didn't necessarily defend the company, but some of their complaints to me were a little bit dumb. But there are some interesting things about the shipping. So we do see priority mail. This was priority. Now I know what you're thinking. You're thinking, is this a photo of something else? Like this is a photo not related to the shrimp. Like they sent them a box of wood shavings. No, they used wood shavings as the insulation for shipping the shrimp. Now I personally have not encountered this before. I've never encountered shipping shrimp or anything in wood shavings. I don't think there's necessarily any reason. Well, technically it's bad. Besides the fact that it is probably very messy and not the most enjoyable experience to receive mixed as for food, but it probably insulates well. I've been trying to think of a way to shred cardboard in a time saving manner to use it like this because I think that'd probably be a good way to get some insulation. Right now, I'm kind of stockpiled on insulation type stuff though. For the holidays, I save all the wrapping paper. So for the next few months, when I start shipping again, everyone's going to be getting wrapping paper stuffed in all their boxes. All right. I recycle a lot of shit, but I've not ever thought about just sawing down to 2x4. But maybe that could be an effective way. Maybe if you bagged it, it would probably not be as bad. If you put this in bags, ooh, and like that, like bro, imagine bagging fish bags and heat sealing them with the sawdust inside, and you get the yeah, man. Maybe that'd be a better. Anyways, I'm talking about this sawdust too long. But that's one thing, the sawdust. But let me read this actual post. Mick says, I have insulated such with styrofoam. I think it makes sense. A little bit messy, but it's not that bad. It's not actually that bad. So, this is the post. I don't know if anyone else has had a bad experience, but they sent my shrimp without a heat pack. All right. They used wood shavings instead. Thankfully, the shrimp are alive, but the water is lower than 55 Fahrenheit. I'm in Illinois. I would never expect during the coldest month they would ship without a heat pack. But maybe I'm wrong for ordering in the winter. I don't know. Kind of pissed these poor babies had to even go through that. 1 to 2 days shipping, ordered on December 10th, and they arrived today on December 19th. Now I'm curious just your impressions coming away from that. If you're in the replay, leave your comment what your impressions is coming away from that. Here's my thing. First of all, key note in the second sentence here, well, third sentence here, the shrimp are alive. So we got a negative post on receiving all alive shrimp. And in a comment below, they actually say they ordered 10 shrimp but received 18 shrimp. Now, I'm not saying that just because the shrimp arrived alive, you forfeited your right to suggest better shipping practices, but it does just come across a little like, really, bro? You got like 8 extra shrimp. They showed up alive. Like you're gonna make this whole post complaining. Now the shipping delay is huge, but as we all know, got no control over that. Like a 9-day shipping delay, even if there was a heat pack in there, there's no 9-day heat pack. Okay, so at that point the heat pack is irrelevant. Second of all, I would probably ship the shrimp without a heat pack in these conditions. I'll be honest, myself. Like the isopods and the shrimp, I really don't use heat packs for. Um, I ship them nicely insulated. You know what I mean? And I watch the weather, but I would never put a heat pack with them because it's a waste of fucking money. And also, I like using the smaller containers, and it's kind of annoying fitting a heat pack. I'd rather just add more insulation. You know. Now, I am starting to discover, as I'm shipping more, that some fulfillment centers are fucking horrible, and they will leave packages outside and things like that. But a lot of paths, I need to do some tests and leave a thermometer in. But I really don't think some boxes get super duper cold in transit. I think some boxes that they actually are traveling in relatively good conditions if they're on time. Where you get into trouble is if they're not on time, they don't get picked, put on the truck. Someone's lazy, they leave it out somewhere. It's stuck somewhere over the weekend, things like that. That's why shipping as far away from the weekend as possible is ideal. But already I feel like this person is complaining about weird stuff. But the wood shaving thing is kind of like my one main question. Like, is this acceptable? Because I prefer this over people will put goddamn fiberglass, like the insulation stuff. I've gotten some things with that, like loose, and I think that's like terrorism. That's like bioterrorism, bro. Don't send me that shit. Like that pissed me off. I get fucking mad when I open a box and I see that shit. I've had it's mostly people like Aquabid. Like I don't think there's really that many companies shipping things with fucking fiberglass exposed like that, or like the little insulation that like it's all itchy and stuff. That's near Carradine and a tub outside till 45F, no problem. Yeah, them and the isopods both see you can overwinter most places as long as they don't freeze solid. It seems. Now, looks like again this person is an uneducated individual when it comes to shipping, which somewhat I do understand because it is hard to no one when you're just a customer, no one explains to you what these things mean. Like, okay, you sent the order December 10th, but they didn't ship it until whatever. Because shit, I get behind on some things. You might be like, bro, is it in transfer for 50 days? No, they haven't gotten it yet. So here, it was accepted at the USPS facility on December 15th. So it was actually only in transit for 4 days. And if I look at the calendar, I have my suspicion. Yep. So this is my suspicion. My suspicion seeing this was that December 10th was probably a day, like a Wednesday or Thursday, and they just happened to order when they missed the shipping window. And when you look at the calendar, December calendar 2025, we'll see that the 10th is a Wednesday. So depending on when they ordered, if it was busy, maybe they ordered at the beginning of the day, but for whatever reason they weren't able to fulfill their order on time. Maybe they don't ship Wednesdays, but most people ship Monday through Wednesday for 2 day shipping because it's going to be delivered all the way up to Saturday. So usually you're good. I have sent some things on Thursday and risked it sometimes if it's closer because usually it makes it by Saturday. But basically, they ordered it on the 10th. A label was purchased, but it didn't get fulfilled, and these people probably don't ship Thursday, Friday, Saturday, or they shouldn't. Then you go Sunday, and then Monday. So they couldn't be shipped the day they bought them, and the next available day that they were going to be shipped was Monday the 15th, which is when they were accepted at the USPS facility. George said, classic Reddit moment. It's like Amazon reviews that leave negative reviews for packaging arriving later than ETA. Yeah. Okay. So, yes, this person this poster is a complete fool's clown, a buffling buffoon. Um, Now, where did I comment? I know I got downvoted. I said, wood shaving aside, 55 degrees Fahrenheit is not close to being a problem for many shrimp. They may have taken a look at the weather and determined a heat pack was not needed for the path the shrimp were traveling. And then this person said, that's a bold call for somebody shipping to Illinois in December. Exactly what OP said. 1 to 2 days shipping ended up being 9 days in the mail. You have to account for that if you're shipping livestock. And I said, I mean, from what I'm reading, all the shrimp arrived alive, and OP even got more than what they ordered. So maybe they did account for it. Like they should have accounted for XYZ. I'm like, well, all the shrimp arrived alive. So clearly they we have no reason to believe they didn't account for it because nothing went wrong. And now that we look at the history, there was a delay. It was shipped on a Monday, and it was. I hate this. This is the worst. In transit to next facility. I hate. This is where like packages go to die. You're like, I don't know when it's gonna finally fucking get somewhere. But it was in transit to next facility for 3 days. They arrived on the 19th, so double the 1 to 2 day thing. Although a lot of them priority is now more like 1 to 3 day. I think it actually says 1 to 3 day, depending on where you're sending it. So yeah, 4 days, not 9 days. A lot of people here can't read, have no idea what they're talking about. It's a travesty, man. It's an absolute travesty. Zebra Aquatic says, my former neighbor kept cherry shrimp year-round outside. Indeed, that is what I hear. I'm like, what's the most controversial post? Can you sort by controversial? In a total? No, it's not letting me sort by controversial. Come on, man. What's going on? I want to know what the controversy is in the aquarium subreddit. Tanks. There's a beta post that I'm looking for. Yo, I just went to tanks. What am I talking about, dog? Why am I going to literally just tanks? Like the machines of war tanks. What else did I think r/tanks was going to be? We've got a new hobby, heartbreak. Nick said, to be fair, I don't understand people getting mad if package arrives a bit late as long as shrimp or fish are okay. That's what I'm saying. I don't really understand. Now I thought this was interesting just because of the dimensions of the tank they put all this in. And I can only conclude that this is why talking about gallons and stuff. Like, look, we're making progress in the hobby. People are starting to talk more and more about the stuff that actually cares to a degree. But this person bought a 10-gallon tall tank. And before we even get into what's going on with this post, let's see what they we have stocked in here. So we got like a gold dust molly situation. We got some kind of looking like a whiptail catfish with maybe a bit of some white patching going on here, or maybe even this is like one of those anchor cats. I don't know. Is that a weird angle? No, I think it's a whiptail. That's a Pomba scape. No, we got cherry red shrimp. Okay, this looks like a nice red shrimp. Karen Haas says Banjo Cat. Thank you very much for that. See, there's always someone that actually knows what they're talking about. Okay. Okay, we got another like a Molly Platy situation up here. Then we got this sad little fish that probably is dead at the time of us reading this. And maybe this is either a small mono or some multi shrimp. Okay, so here's the 10-gallon tall. And they say, I got a 10-gallon tall tank, top fin with lid, filter, bubbler, and heater. I will say, the tank itself, yeah, looks fine. They got a total setup. This is one of those ones where it's kind of hard to stock, figure out what you want to stock with this, with these dimensions. What would you guys put in a 10-gallon tall? I'm even trying to think. I feel like this could be a good breeding tank for some things, like some kind of pair thing that's small, like a pair of you could do a pair of Elisoma, because what I've noticed with the Elisoma, the fry really do avoid the parents. I haven't ever seen the parents like eat the fry, but the fry do like stay very hidden until the parents are out of the zone. But if you had this like height, I do notice that the Elisoma fry, they'll hang out bottom, top, or whatever, basically wherever somewhere else is in their space. And the male tends to defend the territory that he has its eggs, which would tend to be in like the bottom mossy area. So what has been nice in taller tanks is you kind of have the adults hanging out at the bottom, and then if you got some like plants at the top, the babies will hang out. So I think something like that, like an Elisoma or a Scarlet Batis pair. And yeah, Karen said nanofish and shrimp. That's basically all you could go. Um, I don't know. I don't know which nano fish that like schools a little bit or whatever you could get away with in here and not get crucified. But again, the fact that it's 10 gallons, depending on who you're talking to, that might be the escape hatch. Like, oh, it's a 10-gallon tank. What you going to say to me? It's 10 gallons. Oh, it's 10 gallons. We're fine. You know what I mean? Hard to keep most fish in that tank mix set. Kill least killifish, I guess. Chili rasboras. I do think people like chili rasboras, they're so small, man. There's a lot of people that say you can't keep any fish in a 5-gallon tank, like except for maybe certain betas, and people fight you on that. I'm like, bro, there's a lot of fish. There's a lot of fish I think I would keep and do keep in a 5-gallon tank. And I think I would I'd keep chili rasboras in a nice 5-gallon. But then other fish that we have seen, some killifish, yeah, for sure. Some clown killies. But other fish, we've seen these on the one site, the Danianella cerebrum. It was like the smallest freaking fish ever, and they produce a sound. Like this is, I think it's like a 0.2 inch fish or something like that. Like you're telling me I can't put this in a 5-gallon, bro? Come on, come on. Rosy loach, Endostomus. Rosy loaches are super cool. I think ooh, the paradox fish. These are cool. Look at that, man. Let's see, what are they saying the size of this thing is? Bro, I'm like blind. So blind 25 to 30 millimeter. These killifish, which are live bears, at 1 inch. Yeah. Get rid of the pump though. All right. So they added to this tank. We've got a lot of good ideas on what to add to this tank. We've got a lot of good ideas. This is what they added to the tank. The temp is 76 degrees. Let the water cycle for a few weeks before adding any fish. Now, these days, when I first read that, I'm like, great. Let the water cycle for a few weeks. Now, these days, I'm starting to know better to like ask, okay, but like, what did you do to provide the ammonia for the cycle? And I do think without adding anything, I think there is like a certain amount of ammonia that can kind of show up in tanks anyway. I forget how, but like to actually grow a large enough colony to sustain what I think they're about to add in there, you probably need to be adding in some flakes or something like that, just something to get the cycle going. So I would have to ask that question. But this is what they added to this 10-gallon tall: an aquatic frog that disappeared the first day. Got the water tested. They said it was absolutely perfect. Added one molly, one pleco, and two guppies, and three shrimp. All died within 2 weeks. Shrimp are great. Did have a blob with eyeballs that I suspect was a baby and did a celebration dance. That poor thing got stuck on my filter. I since added a sponge to it. All fish died laying on the bottom. Tested the water again. All good. So this is sounding very suspicious. Waited to add fish. A coworker gave me a small catfish, so that's what Karen identifies the banjo catfish, one platy, and one guppy. They all died the same way in about 2 weeks. Catfish is suspect, still alive, but very elusive. Shrimp are doing great and had babies. Happy dance. I feel like this is doomed to be a shrimp take at this point. Changed 20% water and cleaned. Got the water tested again. Good. Did they test the water after the water change? Waited 1 month, water tested again. pH was tiny elevated. Add a tank starter. Ooh, excuse me. Add three guppies and a platy. just draws at random shit here. 3 days ago, everyone looked great. I'm surprised the shrimp did so well with the new tank. Yeah, I mean, this shrimp looks great. It's just happily everything else is just dying around it. It's like catastrophe, plague. Man, this thing looks like happy as a clam, man. And it's got red on the legs. It's a good looking shrimp. I think this person got to take the hint. Just let the shrimp win. Today, one guppy laying on the bottom. I'm heartbroken. I feel so defeated. I read everything I can, follow every thread here, and ask a million questions at 4 different aquarium stores. What am I doing wrong? Sad fish mom. Now, what I thought was interesting about this post, besides the fact the challenge a 10-gallon tall could bring, is it's got so many things of like the typical like newish fish keeper in here. On the positive side, this person does seem to care about the fish and want to do better. Karen says, I wonder if the water is too soft for guppies, mollies, and platies. Yeah, that could be it. That could be it. Although I do wonder what happened to the frog. Cause it said the frog disappeared. So like looking at this tank, it's first of all it's hanging off the side, which I mean I wouldn't really worry about this too much. I'm not saying that I think, but there is a crack here. There's a crack here, and if the frog somehow could jump out, which I've never kept the aquatic frogs before, I feel like she probably, if she moved this, there would probably be a crunchy frog. Karen said the frog escaped. Yeah. Now there is a lid on here, but this is a huge gap in the back. I'm seeing like I don't know what kind of we gotta look at top fin 10-gallon tall. Okay. Yeah, this looks like the one. I'm trying to get a better view of this lid. because I'm trying to see if if the lid just has a gap, or if they have no. You can kind of see a similar gap back here on the back. So they left a lot of space for this hang on back. Maybe more space than needed. So here we go. What catfish do they have? Karen said it looked like a banjo catfish. Okay, so they got the hang on back, and we got the lid. Yeah. Now in this video, they're not really showing the back. I think that just goes to show they probably know it looks horrendous. But I will say in this photo, the lid is clearly not all the way on. They got the airline tubing run through over here underneath it. The filter looks off in the pic. Yeah, I don't know what's going on in the pic. Mick says banjos can eat others. I mean, it wasn't that big. What do you think? It was like eating slime coat or something? Like, this is the photo of the fish. If this fish itself doesn't look like it's in perfect health, I would say. Does seem to be munching on some kind of plant there. So feeding it some plant matter. But the other thing that's interesting about this, yeah, you got someone that cares about the fish, but this is the thing you always hear. This is the line that always kills me with these people, man. And I'm sure I've been guilty of it myself at some point. But I read everything I can, follow every thread, ask a million questions. And banjo cats prefer sand to burrow in. Okay, yeah, this definitely don't look like the most advanced substrate. They always say I follow every thread. That's like I've read everything. I'm like, well, if you read everything, the dimensions of this tank probably, like if I was starting off, probably try and do a better dimension tank. That would be first. If you read everything, maybe throw some plants in there. You know what I mean? If you read everything, like Karen said, like some of these fish that prefer sand and stuff, you know, maybe you should have some different substrate. Like if you read everything, then this tank would look different, I think, you know? So it's conflicting because I want to give people credit for caring because I think that is important because a lot of people just don't care that much. But sometimes it's the people that do care, it's even more frustrating because they care, so they feel like they are doing the best they can and all that. And they kind of miss that they are not, they're not, even though they feel like they think they're researching well, they're not. You know what I mean? So then it's harder for them to find a solution. Uh, I've had the lights on a little too long, so I'm switching the fish food for ammonia hasn't worked well for me. I think it makes the water full of pathogens and more fungus. Yeah, I think well, kind of like when you've had tanks for a little bit, then you know it's not like I've ever recent in any recent history thrown had to really cycle a tank in that manner. I think using like snails and stuff is probably easier, like just some like pond snails, things like that. Or I guess you could use the ammonia, just the pure ammonia stuff. You gotta do something, right? All right. So we've had our discussion. Here's the best part of reacting to Reddit content, seeing what other people out there think. So here's the top comment with more upvotes on the post itself, and this got almost 400 comments in 2 days. It's relatively decent for this. Mr. Catechus comes by to say, you're missing the sponge and the rest of the tube on your filter inflow for once. Well, the person did say that they added the sponge after one of their fish were killed by the filter. For 2, describe how you cycle the tank, please, if others said. For 3, I'm pretty sure that's a whiptail catfish, not a pleco, and will absolutely outgrow that tank. Then the bottom, look, we got Karen's validation. This person responded, the catfish looks like a banjo. And this person said, too small of a tank for that fish. Okay. And then look, here we go again. We're on track. We're on track here. Definitely a banjo. And he looks injured. Probably from trying to burrow under the decor and rocks. Sand is a must for these guys and a bigger tank. They are not small fish. So I think that is that so far we're on track with identifying some of the problems. Let's see. Next person says this post is heartbreaking for different reasons. Sounds like you didn't cycle a tank. Running for a few weeks in the cycling, you need to help the bacteria colonies along. Yeah, I think this is what I think. Adding just bro, add some plants when you're setting up the tank, man. I think that kind of helps a lot too. But but just filling it up with the gravel and these structures. I always feel kind of weird with the decoration that's like some dead bone skull looking thing. Like this kind of feels like an omen for what's what the fish are about to face. Like, hey man, welcome to the tank. Hope you enjoy your stay. You're gonna be looking like me pretty soon. Like I don't know, man. Okay, it said they add fish too fast. Bacteria can't handle it. Google sizes for the tanks. This person is saying ghost feeding for simulating the bio load. Man, this person, don't listen to people at the store. The fish they're selling you is not good stock. A pleco will wreck a 10-gallon. And most of the other fish need to be in groups of 6. Here we go. I'm honestly impressed the shrimp have lived through this. Usually they're the first to go. This is kind of interesting because the second comment is I'm also very confused how the shrimp could thrive, but no fish can. Very opposite of my experience in aquarium keeping. It's interesting because shrimp do badly in new tanks, but like overall, shrimp are, they call them like water cockroaches. They're really hardy. People still kind of see Neos as being super sensitive. Every time I go in these Reddit threads, like people do still see shrimp as being like more sensitive than I think they should get credit for. But in this situation, it's kind of interesting. The shrimp are doing well. They're probably loving to eat off all the dead fish. Your banjo catfish would need a place to bury itself comfortably. Okay. I didn't realize what he is. See, I read everything. I ask as many questions as I can, and yet they did not have the ID of the fish in their possession. They added to this thing. You see what I'm saying? Like these people that say they care, they ask all these questions. It's kind of like I get it, man. I'm glad you care, but like, bro, you didn't even try though. You didn't even try. Knows how to use Reddit, but not Google Lens. Have you guys used Google Lens for? Have you used Google Lens for fish a lot? I've never tried it for aquarium fish. Is it actually any good? Because I would be a little bit suspicious. Never seen that plastic decor in nature. You're in nature a lot. You're in nature a lot, Mick. Now we talked about betta fish as a white elephant gift trend. I just wanted to give a little bit of an update. All right. This is what someone had received the betta fish in. Now, the most interesting thing about the white elephant gifted betta fish is, I think, the containers they were gifted in. I feel like you could tell a lot about the people that gifted them. At least some people did put together a kit. Now, it was a critter keeper kit, most of them, but some people did at least provide a kit of aquarium related things, an aquarium related container. This person put the betta fish and a handle of Tito's. OD. Yeah, man. I feel like a picture's worth a gazillion words. And you just got a mental picture of the absolute Neanderthal that pulled this one off. Anyways, this person took this and gave the beta this fish tank, which I think is a pretty nice tank for someone that is just starting. Got the moss little coconut hut here. Got some scattered plants, heavily planted, right? Heavily planted. Got some interesting wood here for a betta fish. Yes, great first tank. Good job. 10 gallons, heater, filter. See what the flow is like on that thing, but this betta does look like it's got relatively long fins. But besides that, sure. Let's see. The betta fish subreddit has to have been going through it in the last week or so. It has to. I'm gonna just have to Google. I'm just have to search white elephant in here and just see what's been going up. Okay. Yeah, we got some white elephants. My class decided to do white elephants, but I never expected this. We're at the end, and the boys were demanding us to be careful with the package. Turned out to be a live betta fish. I was the only one not laughing. Jesus Christ. I was the only one not laughing. What's interesting, they asked for help on how to take care of the betta fish. Here's some of the recommendations, some of the usual things here. What's kind of interesting is this recommendation had Good Fish YouTube channel or podcast. I always think it's interesting to see what they recommend. They recommended, although they spelled recommend incorrectly, so maybe take this recommendation with caution. Watercolors Aquarium Gallery, Keeping Fish Simple, and Aquarium Co-op. I think it's interesting. I don't know how popular the podcast Watercolors Aquarium Gallery is. I found it because I was looking up aquarium podcasts. And it's like one of the only ones on the podcast app that pops up. But I thought it was cool that they're on there first for what the recommendations were. People need to learn about nature. The term aquarium fish needs to be annihilated. Fish comes from nature. Okay, but Mick, we're talking about. I feel like betta fish, like the domestic bettas, probably do earn the title of aquarium fish. Like these are not. These are not like this is not a creation of nature. This is an abomination through like 1,000 years of domestication, selective breeding. So I feel like the cup bettas are probably aquarium fish. You know what I mean? But no, I feel you. I do feel you. This is a different white elephant gift. 4 days ago. This is another one 6 days ago. Victim to white elephant. Damn, bro. The bettas in these cups look absolutely jacked. Yo, I will say it's kind of interesting. Tannins are definitely like, bro, tannin. Everyone is the tannins, the tannins. You see this new tank? It's just like, boom, tannins. Message received. Looks like you put them in some kind of bath. This looks like you put them in some kind of medicine bath. It's blue. It's both an introduction post and a dangerous care post. Okay, interesting. Look, 11 days ago, better from White Elephant Party. But see, come on, Critter Keeper, this is a step up. This is a step up. At least like in the cup, you kind of got to deal with things a little faster. You know what I mean? At least there's more volume of water. Now we got the Critter Keeper with the pothos, and then into the tank. We don't got a photo, but 3 days ago, white elephant. Oh, this is good. 0 upvotes but 16 comments. I was gifted a betta as a white elephant. I've been spending the last week setting up my betta's tank. I've changed the water 4 times since I started the tank and added my betta into the tank after a 6-hour cycle. What does that mean? What is a 6-hour cycle? I don't know what that is. What is that? 6-hour cycle. I know you're supposed to go longer, but you look miserable in a small container for pleasure. No, right decision. If you just got gifted a white elephant betta, don't wait for your tank to cycle to put it in. It's one betta, like one betta in the 10-gallon. Like you should be able to stay ahead of that. You should not. You should be able to keep the 10-gallon without going crazy. Throw some plants in there, yeah, yeah. One betta and a 10-gallon, it's gonna be fine. It should be fine. Do water changes on it every day if you need to, if it's not cycled, but I, you should be able to keep it alive. Nick said 6-hour cycle problem solved. I think it was the right decision. I think it shouldn't really be a problem. I added live plants, a water filter, thermometer, blah, blah, blah. Seem relatively happy, and the biggest hiccup I've experienced is lack of eating. And I got him as a gift. I was given a bottle of small betta pellets, but he will not eat them. What I've learned is probably due to the amount of wheat in these pellets. So I got him a container of bug flakes. I tried plenty of those, but he chews and spits it out. Okay. Now, as we evaluate the condition of this betta, I think we're all thinking one obvious thing that seems to be occurring. But I just want to talk about if there's any comments about the cycle. I think any comments about the cycle is like a waste of time. You got gifted a betta, it was in a cup, you put it in a bigger tank. Great, right decision. Waiting for it to cycle, shut the fuck up. You can change the water every day, but honestly, realistically, throw more plants than this in here. But one fish not being fed, very key word, not being fed a lot of food, you should be able to stay ahead of ammonia problems in this scenario. You really should be able to. But this is a new person, so probably not. Um, but I think this is interesting because the white elephant gift thing, I'm sure some people in the industry, and even if you talk to some people, like even at clubs and stuff, people are conflicted about the goldfish carnival thing because while it is like bad or whatever, it's like everyone's introduction to fish tanks. So it creates new hobbyists. It's one of the biggest places people get into the hobby from is that goldfish at a carnival. And this white elephant thing is like kind of similar. It's like you gain a better fish at a white elephant gift. This trend could be spawning like a lot of new future aquarists. But and I don't know if you say like okay, but the fish are going to suffer. Honestly, like for a lot of fish, probably suffered while I was learning how to keep fish. So that's one of the things about keeping fish that is kind of conflicting in general. Is like for everyone that's kept fish for a long time has killed a fish or two at some point. You know, everyone makes mistakes. Also, fish do die, but I'm just saying, like everyone through some kind of mistake they've made, I would say has accidentally caused the demise of a fish before its time. So, is it really that different in these kind of scenarios? Like again, a lot of people's first story is getting the goldfish at the carnival, and maybe some people learned how to take care of it better, faster than others. But ultimately, they became like really great fish keepers, maybe even owners of fish keeping businesses that maybe are improving the hobby and overall welfare. That yeah, I'm overthinking this quite a bit, but you get what I'm talking about. But on the flip side, this is the fate of some of these white elephant bettas, even for people that are actually trying. They got this guy in the damn the wave maker, man. He's in the like at the Great Wolf Lodge. They threw him right in there, bro. Right in there. This guy he's getting thrown all over this damn tank. He's getting thrown all over. I would say one thing that's interesting about the cup bettas, man. You always see a betta. Like, I haven't really seen a betta that looks exactly like this before. These things all just look so different. Bob said, "Thank you, Nick. Your stream motivated me to do late night terrarium work. Glad I could help. I mean, that's what it's all about, man. That's what it's all about. Get some shit done." So reduce flow, remove. I love I love Reddit people. Okay, you see this tank. What do you think of that? That should immediately be done. This is my list of what should immediately be done. Add more. You could add more plants, and the filter. If you can't adjust the flow of the filter to be low enough, then you need to switch the filter out. That's literally the only two changes that I would recommend at this point for this person. Add as many plants as you possibly can. Maybe you could add a piece of wood, but honestly, like I'm just keeping it simple. Add more plants. If you reduce the flow, if you can't get the flow lower with this filter, then get a new one, and that's it. All right, change the water regularly. If you want to do some tests and stuff to help guide when you're changing the water, you can do that. But that's where I keep it. I love Reddit, man. Reduce the flow, remove the stones. Parentheses. Replace with aqua soil. Bro, this is some NPC just absolutely asinine advice right here. First of all, this person's new. They're setting up this tank. The last thing they need to be doing is replacing the fucking substrate. Okay, like, can we at least let's get the filter situation sorted out? Like, can we prioritize here? Second, you're gonna suggest replace with aqua soil. You're not even gonna suggest which aqua soil. Because what if they choose one of these aqua soils that actually are um, releasing ammonia? All right, let's go to 2 Hour Aquarius, man. 2 Hour Aquarius got some great information. Many aqua soils enriched in ammonical nitrogen. All right. Good quality aqua soils would significantly deplete your nitrogen source at 6 to 12 months. Okay. Let's see. Although ammonia levels were depleted over time, HOs were still okay. Okay. Yep. Let's see if they talk about new ammonia-rich aqua soil makes it okay. 2 Hour course. I'm trying to see if 2 Hour course page on ammonia uh, says anything about if it leaches into water or not. But anyways, my point is I hate this. This is someone that has no idea what they're doing, and you're like, not only are you saying replaces the rocks, which I get it, it's the ugly rocks. Okay. So a couple fish says yes. That's what I'm saying. So not only is this a new tank, it's like emergency situation. It hasn't had a cycle or not. We got the betta fish. He's got the Beach Boys playing in the background, surfing, surfing. Like he's fighting for his life over here. Okay, I'm glad you addressed that. You reduce the flow. Your next thing is, let's get rid of the rocks. Let's just fucking stress this guy out, remove the substrate. Maybe it's already started to get some bacteria on there. I don't know. And then instead of being like, you should add a inert sand, an inert gravel, it's not painted. Replace with aqua soil so we can really extend this cycling process and, perhaps expedite the execution of this betta fish. But I love people on Reddit, man. And this got 21 of us. I'm downvoting this. This man's a fucking fool. This man's a fucking fool. Okay? A fool. Besides the fact that this aqua soil could be contributing to the early grave of this betta fish, why don't you not just suggest like a sand or something, bro? Like something inert for someone that has no idea what they're doing. Why would they ever need the aqua soil in the first place? Uh, okay, no such thing as 6 hour cycling. You'll have to do a fish in cycle. Okay, this is what I've been loving. You're seeing a lot of these anything betta related. They're just posting these graphics. Okay, and what I love about the graphics is they just say temperament of betta fish aggressive, which I get, but to me it's also kind of funny because like if I really love betta fish, I would be the one out there like, no, Don't call all the betta fish aggressive. All betta fish are different individuals. That does, blah blah. They're just like, nah, they're all aggressive. They're terrible. These are the pit bulls of the fish world, which I get it. What's interesting is though they do talk about like live foods and freeze dry frozen foods a lot, which is interesting. Beginner friendly plants, we got duckweed, Ludwigia repens, and Anubias was the beta suggestions. Here's the tank mate suggestions: Pygmy corridors, Harlequin rasboras, snails, coolie loaches, and shrimp. I mean, I guess I don't have an issue with that list of. I don't know what the my grandpa had the Harlequin rasboras, but I never kept them with anything like better fish to know if they were like huge fin nippers or not. So I can't really comment on if they're a good suggestion. We have sororities. Sororities are a group of female bettas should only be attempted by expert fish keepers. Never keep multiple males or male and female betta fish together. I guess that's probably decent advice. Okay, a lid, a filter with low flow, a heater, lice, decor, tannins, air gap, plants, and substrate. What's interesting, sand, gravel, or if you have plants, aqua soil topped with either sand or gravel. People get really mad at capping aqua soil. I've been hearing a lot of people mad at capping aqua soil recently. And I guess I kind of get it. Like, yeah, I guess you could just do sand with root tabs at that point, right? And it would be more economic. Okay, water changes, nitrogen cycle. This is interesting. Healthy weight diagram from the top view. Skinny, thin, ideal, heavy, and obese. Interesting, Will. Interesting. All right. I know how to keep a better fish now. Okay. This person seems like they left a decent comment. More like plants. Nope, downvoted. Downvoted. Talk about switching to aqua soil. Shut the fuck up, bro. These people don't need aqua soil. Come on. What's going on, bro? They've been so marketed to. This is crazy. All right. Yo, this is crazy. Okay. 11 days. This you could tell that there was quite a lot of betas given out. Help got better during White Elephant Party. He's got the 10-gallon tall tank as well. So is that the one of the more available kits? Like, is there a reason that's what these people are all buying? Look at this absolute beginner help, please. Man, this better already look like it's ready to die. It's over. It's over. That's why I said you just got gifted a hospice patient with a bad attitude. Ole. Okay, we got the better fish graphics. God damn it. 12 days ago, it's just help in the picture of the test results. I'm not actually seeing anything bad on this test results. Yo, what is this post? It just says help, and it says, how do I fix this? This is the test results. What's the problem? We got soft water here. Looks like low GH, no chlorine. It looks like it's got some low KH. Look like it's got like a 6.8 P. Yo, this water is looking peak. This looks great. No nitrite. What's the problem? Yo, what do you want help with? Is this a test for your source water or the tank? I tested my tank water. Is it bad? Question mark. Yo, everyone, look. If you're on the replay, if you're on live, I would like us all to have a moment of silence for the fish that this person is about to ruthlessly slaughter over the next few months. Okay, can we all just hold up? All right, man. Yo, this ain't good. This ain't gonna be good. This ain't gonna be good. If you're winning the fish owner lottery and you're the fish and you're on Reddit in your fish tank and you say, "Oh wait, wow, my owner posted this post." It's just hell. It's a test of what seemingly is perfectly fine water, and they say, "I tested my tank water. Is it bad?" Oh man. Oh man, this is not going to be good, man. It's acidic, which is probably causing your tank to not cycle. Jesus Christ. we got the blind leading the blind, man. It's like 6.8, bro. It's fine. It's fine. We don't even have a. First of all, we can't even say. We have no reason to believe it's not cycled. They haven't told us anything, and this doesn't seem to have a nitrate test on it. Oh wait, no. Here's the nitrate test. I'm a fucking idiot. Okay, there's no nitrates, but like we don't see the tank, so there may not be no reason for nitrates to be in there. Yes, it was not planned at all. It was a white elephant gift. Okay, we got the graphic again. Yeah, bro. This white elephant thing is crazy. Wait, is this the same person posting help? There's so many things. Just yeah, this person posted multiple posts saying help. Yo, this is okay, man. Yo, God bless that person, man. God bless you. God bless him. It's a nut 7 days ago. White elephant gift. White ends on the end of the tail. Man, I want to buy some like embellis, some acanensis, some wild type of the splendens complex, but I got better as a white elephant. Man, pick up the fish, but that's where you need to pick up fish so we can see the situation. Mick, shut up, Mick. That will make no sense. What are you talking about, Mick? You need a picture of the fish, situation. Like, yo, what? Turn on your telepathy, bro. Like, can we do some astral projection? Like, stop being so lazy, man. You've got the tools to help this person. You're just making up excuses. I don't know what to tell you, man. I don't know what to tell you, bro. You gotta do that remote viewing. Like, bro, can you just be helpful? Ooh, look at this. I like this tank. Look at this low, shallow. Look at that. All right, I gotta get off the stream, but before I get off the stream, I just gotta say I'm waiting for my peppermint pikes. Okay, waiting for my peppermint pikes. The Luciferus aura. Waiting for those. Where they at? Christmas just passed. I don't fucking got any. Um, I'm waiting for my UNS livestock holding system. I don't know where Christmas passed. I don't know where this is. What's going on? Universe? How's it going? Besides that, I don't really have much to say. Hardest working. We're gonna keep rocking it out to the year. Nick said you had to order the peppermints. No, I haven't. I have not ordered the peppermints. But 2026, we got to get the peppermints. That's the goal. But no, it's been a good time. It's been a good. Oh, yo, what's up, Big Steve? I appreciate you. God damn, why are you still awake, man? Go to sleep. George said, "I'm here. Let's rock." Yo, George, you gotta be trolling me right now. You're trolling me. There's no way that you perfectly come in every single time when I'm about to get off the stream, George. I know you're trolling me. You've been exposed at this point. I think you've been here. You've been here the whole time. Now you're just showing up now in the chat just to mess with me. What's up, man? I hope you had a good Christmas, George. Okay, I'm getting off, but we'll be back soon. We're closing out the year strong. Okay, hardest working next year. I'm hoping to improve on all my personal flaws, all my content flaws. We're gonna keep being the hardest working fish trail next year. I'm trying to be the hardest working fish YouTuber next year too. That's gonna be, yo, I don't know about that. But take care of yourselves. Enjoy the rest of the holiday season closing out. I know some of us, we got some work to do tomorrow. I know I got a lot of things burning a hole in my head, and staying up till 3:30 a.m. probably ain't gonna help. But holy, what you gotta do? I'm alive. I'm gonna have to keep being alive and trying as long as I am. So, yo, wish me luck. George said I'm gonna stream more as well next year. Good. That'll be fun. That'll be fun. Pop in more George's streams. Come on, people. Come on, people. Here's a link to George's account, man. Tap in. Tap in with George RP3. He never comes onto my live stream until I'm getting off, bro. More fish? Yeah, more fish. I don't know. Tomorrow could be my last stream of the year? Question mark. Tomorrow could be my last stream of the year. Jorge asked if I was available to stream on the 30th. I might be streaming with Jorge tomorrow. I'm not sure. No, wait. Well, yeah, today's the 29th. So, yeah, the 30th, which is Tuesday. I'll probably not stream today. Yeah, I'll probably not stream today. More aquariums? Yo, I like what you're talking about, George. And again, thank you, Big Steve, very much for the $2 donation. Hardest working. I really appreciate you. We'll be back again at the very latest on Tuesday. And perhaps that would be the last stream of the year because I'll probably be busy on New Year's Day, just hanging out with Jessica and all that. And then it's off to the races, man. We gotta get shit going. We cooked up a lot of things in the last half of the year. Club president trying to pull off the swap. The event seemed relatively successful. I think the board's meeting tomorrow, so we'll probably talk about stuff we're gonna improve and things like that. But we got some stuff breeding. We did not get to the setting up this fry tray, man. We did not get to setting up this fry tray. But I'll probably just have to do that off stream, man. I'll probably just have to do it off stream, and you'll see. You'll see when it's got eggs and stuff in it. But yeah, I'm a hop off. Take care, everyone. Replay crew, don't be a stranger. Leave a comment. Share your thoughts. Thank you, George. George, I hope your coming year is full of blessings for you. I appreciate it. Hopefully, I could use it. I just say, to describe my life right now, how I feel like I'm in hell. That's how I feel. So, look, appreciate you guys very much. Take it easy. This has been In My. Do you ever feel like a banjo cat in a glass box full of regret? Do you ever feel like a better fish that was given as a white elephant gift? Do you ever feel like a RoboCop looking for food and the search never stops? Do you ever feel like in my tanks and I am out? But first I gotta deny the light and let it shine. Yo, this side view is crazy, bro. I am ugly as sin. All right. Am I